I request to discipline committee in charge to please maintain the decorum and proper assembly of students in main auditorium. We are going to start our day second ISFCON-2025 technical session 6. After all this session, we have valedictory function, and in valedictory function, we have chief guest, Dr. Professor Felicitation, and all results will be announced in 12 categories in the valedictory function. Thank you so much. I request Dr. Ankit Mishra sir, and I also want to share one proud moment with all of you that yesterday we have 12 category oral online sessions, and in that we have more than 270 presentations, and it's a proud moment for all of us. And today we are going to start in the afternoon session oral offline and poster offline session. Hello. So we have one dignitary among us, Dr. Ankit Mishra.
Thank you so much. Good morning everyone, I am Dr. Ankit Mishra from Pharma. We are conducting one quiz competition in this international conference, which is very simply based on knowledge of pharmacy. We have taken one permission from Dr. G.D. Gupta sir that we are going to give 3 prizes in this category. First prize will be 5,000 rupees cash and a trophy. Second prize will be 3,000 rupees cash and a trophy. And third prize will be 2,000 rupees cash and a trophy. This, thank you so much sir. ये काफी इजी क्विज है जिसमें अंडर ग्रेजुएट लेवल के क्वेश्चंस पूछे जाएंगे. आई एम रिक्वेस्टिंग कि इसे केवल स्टूडेंट्स तक ही रखा जाए बिकॉज फैकल्टीज आर टू मैच्योर फॉर दिस टेस्ट. तो बहुत ही सिंपल सा टेस्ट है. हमारे ऐप जिसपे टेस्ट देना है उसके अंदर बैच कोड हमारे जो नोटिस बोर्ड से बाहर वहां पे लगा हुआ है. साथ में जो स्टॉल एक अवेलेबल हुआ है वहां पे भी ये प्रेजेंट है. इसका टाइमिंग 11 AM से लेकर के 2 PM तक रहेगा. इसमें सभी को क्वेश्चंस ओके ठीक है सर. 12:30 बजे सर ने इसका टाइम अलॉट किया है. 12:30 बजे से लेकर के 3 बजे तक ये टेस्ट एक्टिव रहेगा. इस टेस्ट के लिए आपको जस्ट एक ऐप डाउनलोड करके मोबाइल नंबर से लॉगिन करना है एंड यू हैव टू जस्ट अटेंड द टेस्ट. बस इतना सिंपल है. क्वेश्चंस रैंडम आएंगे. सभी स्टूडेंट्स को अलग अलग क्वेश्चंस मिलेंगे और उन क्वेश्चंस को आपको सॉल्व करना है. बाय 2:30 वी विल अनाउंस द रिजल्ट. हम 2:30 पे इसके रिजल्ट अनाउंस कर देंगे. आप में से अगर किसी को भी कोई कंफ्यूजन हो तो यू कैन कॉन्टैक्ट मी. आई विल बी अवेलेबल देयर. मैं इसमें आपकी जितनी हो सकेगा हेल्प करूंगा. बाय 12:30 to 3. थैंक यू सो मच.
Thank you, Dr. Ankit, for this announcement. And within few minutes, we are going to start our today's function. ISFCON 2025 ceremony of ISF College of Pharmacy, where you will witness our students' achievement in academic research and overall personalities in the field of pharmacy profession. Let's begin this auspicious day with our day second 6 technical session. For this, I would like to call on stage Dr. Akhilesh Singhai sir as the Chairperson, Director, LLCT University. Sir, please. Next, I would like to call upon stage our worthy honorable guest, Dr. Pooja Chawla, Professor, Baba Farid University of Health Science, Faridkot. Please, a big round of applause for Dr. Pooja. Now I request our worthy director, Professor Dr. G.D. Gupta sir, and Principal ISFCPR, Professor Dr. R.K. Naran sir, to please present a floral welcome to our chairpersons. Thank you, sir. It is my great honor and pleasure to welcome our guest of honor and resource person, Dr. Hina Tabassum ma'am, Scientist D, ICMR, New Delhi. Please, ma'am. Please give her a huge round of applause to Dr. Hina. I request Director Sir and Principal Dr. R.K. Naran Sir to present a floral welcome to Dr. Hina. Thank you, sir. Thank you, ma'am. Now I would like to call Dr. Sant Kumar Verma, Head of Department Pharmaceutical Chemistry and Pharmaceutical Analysis, and he is also the Coordinator of Scientific Committee to further proceed this session. Dr. Sant.
Very good morning, everyone. Firstly, I would like to introduce today's speaker, Dr. Hina Tabassum ma'am. Dr. Hina Tabassum is a scientist and program officer at the Indian Council of Medical Research. She also established a national task force for various Campo Moreso leading development of hospital-based EPA registry across science with over 1,500 enrolled patients to assess disease burden, outcomes, and prognostic factors. Dr. Tabassum has published 16 journal articles and 11 abstracts in reputed national and international journals. Her expertise spans hematology, toxicology, and neurobiology, and she has actively contributed to national health research programs. Her work has been recognized with numerous prestigious awards, including the Best Public Relations Award in 2016 by the Society of Biomedical Laboratory Scientists India. She has also received multiple international travel awards from esteemed organizations such as the Japan Neuroscience Society, IBRO, APSL, FELS, allowing her to present her research at global conferences. Additionally, she was honored with the MCT University of Minnesota Research Excellence Best Paper Award and several Young Scientists International Travel Grants to attend high-impact scientific meetings in the USA, Germany, Poland, and Japan. Dr. Tabassum continues to contribute significantly to medical research, regulatory affairs, and policy making in India, strengthening the country's healthcare landscape through her scientific and laboratory expertise. Welcome, ma'am. Now I would like to introduce our chairperson of the session, Dr. Pooja Chawla, ma'am. Dr. Pooja Chawla is working as a professor at the University Institute of Pharmaceutical Sciences and Research, Baba Farid University of Health Sciences, Faridpur, Punjab. She has more than 20 years of experience in the research, discovery and development of biologically active pharmaceuticals. Dr. Chawla holds a PhD degree in medicine. Dr. Chawla is also produced work as a guest of two reputed journals of medical sciences. She also presented a poster at FIT and other international conferences. Madam has worked in the area of development of TJD derivatives, common derivatives, C-basis derivatives, to make a few, and the discovery of number of medicinal agents. Welcome, ma'am. Now I would like to invite Dr. Hina Tabassum for her session. Please, ma'am. Welcome, ma'am.
Am I audible clearly? Thank you. Thank you. So good morning to everyone on a Sunday morning, and for all the females here, happy Women's Day. So I would kindly like to give my gratitude to the organizing committee for having me here, and a very generous introduction that was given to me by my peers. So I would be speaking on ICMR's role in promoting research in the subject area of nanotechnology. So before I start, I have a small question. What is a nanoparticle? So can I have a small answer from anyone? Because I will be speaking about what are the lacunae, what kind of research that we have funded in past 5 years or so. But because I understand there are students from B.Pharm, M.Pharm, so we have people who are not actually doing PhD. So we have a younger generation as well. So everyone is aware of what is nanoparticle. Any answer? Anyone? This is a very easy question for people who are going to develop drug molecules for us in the future. So, what do you understand by nanoparticle? What is nanoscale? Anyone? Someone from the first row?
Someone wants to answer?
सुबह पूरी छोले खा के सो गए जल्दी। नाइनोस्केल क्या होता है? मैथ का क्वेश्चन है। साइज में बताइए, साइज क्या होता है नैनो पार्टिकल का? So anything less than
10 to the minus 9.
So anything less than 100 nanometer, right?
Yes.
Fine. So yes. So from me starting with what I want to say. So Indian Council of Medical Research is the apex research body, biomedical research body in the country. We started in the year 1911, and it plays a crucial role in fostering research in India in several subjects, and that includes nanotechnology or nanomedicine as well. When we talk about nanotechnology, it enables precise medical interventions. It is going to revolutionize the diagnostics, drug delivery and regenerative medicine, and that would be more on the personalized mode for human beings. And ICMR actively supports interdisciplinary research and policy development when we talk about integrating nanosciences into healthcare. I would be speaking more on the healthcare part because our mandate is to work on translational research. The translational research is something that can be taken from the lab side to the bedside. So the second slide, nanotechnology and its role in medical industry. So nanotechnology finds its application in several parts of the categories when we talk about medical applications. Most of you must be aware or will be aware about something called as drug delivery. So nanotechnology, the basic research that is going on in the pharmacy colleges across India, the people are working on drug delivery systems. So the drug delivery means you target specific cells or tissues in the body. So you are working on a target specific mode of delivery system. So when we talk about delivery can be of a drug molecule or any biological product with an intention to correct the diseased cell or to take your drug in pursuance of health. So what are the primary features of working on a drug delivery system is you are trying to reduce the side effect of any drug molecule or a toxic molecule per se, because it is a chemical entity, it is a foreign entity to human beings, with the hope of improving the efficacy because you are working towards target specific delivery. Then there is another aspect of using nanotechnology, that would be imaging and diagnostics. So there are contrast agents which can be used in MRIs and CT scans. So this would primarily help you in early detection of the problem. Then obviously one of the hot topics when we talk about use of nanotechnology in medicine is for the cancer treatment, because it is very much required to have target specific delivery or a target responsive treatment for the cancer cells. So there is another field, tissue engineering or infectious diseases treatment. When we talk about tissue engineering, creating scaffolds for tissue regeneration. So this kind of technology would be helpful in the field of wound healing or tissue repair per se, where we have damage to the tissues which might be irreversible in nature by itself. When we talk about infectious diseases treatment, we can have target specific which is more pointing towards killing of a microorganism, because we are talking about infectious diseases treatment. So that would help us in reducing another problem, which is antimicrobial resistance. Nanotechnology or nanomedicine per se also finds its role in development of gene therapies. So nanoparticles, they can help transfer or the delivery of genetic materials when we talk about genetic disorders. And nano formulation, nanoparticles or nanotechnology by itself as a technology finds its usage in the field of medical implants as well. So why use nanotechnology? It might or it may help us in increasing the biocompatibility and durability of that particular implant. For example is any implant for the joints, knee joint, shoulder joint, something like that, or the general dental implant because of the size. So it might increase the durability and the biocompatibility of the product per se. So these are the some of the general uses. There must be many more which have not been covered here. So coming on to the different forms. So this is just a theory slide for different terms that you would come across when you go ahead in your studies. Some of you might take up nano-based studies in your PhD work or so on. So nanoparticles can be divided into different kinds based on their composition, their shape, their size and their surface properties. So these are the broad categorization of nanoparticles based on their physical and chemical properties. If I would like to further divide, they can be inorganic, organic nanoparticles, hybrid in nature or natural nanoparticles. So for inorganic nanoparticles, they can be metallic in origin. Metallic means there can be silver nanoparticles, zinc nanoparticles, gold nanoparticles. There are metal oxides, so titanium oxide, zinc oxide, and there is silica-based nanoparticles. So silica-based, their properties are they are more amorphous and porous in characteristics. And then the latest one in this list are carbon-based. So carbon-based like nanotubes are there, graphene, fullerenes. So this is a kind of inorganic category for nanoparticles. Apart from that, there are organic nanoparticles. So they are polymeric nanoparticles, and they can be further divided into biodegradable or non-biodegradable. So they can be lipid-based. Lipid-based example, you must have heard of it, liposomes. So those liposomes or myces, so they have a base which is lipid in nature. Then there is protein-based nanoparticles. So they can be protein conjugate, or there are nanogels which are protein-based. Apart from that, as I spoke earlier, they can be also divided as hybrid or natural in origin. So natural would be viral nanoparticles. So they can be prepared from both plant as well as animal viruses. Then there are bacterial nanoparticles. So they carry bacterial cells or components of the bacteria as nanoparticles. And then last in this category are the fungal or the fungal-based nanoparticles. So they are constituted from the fungal cell or its components. And from the hybrid means there will be mix of two kinds of nanoparticles. So there would be a core and an outer covering. A core can be prepared from a metal or a metal oxide, and there would be a shell which can be organic in nature. So it can be lipid or proteinaceous in nature. And there are a hybrid nanoparticle called as genus nanoparticles. So they can be two different or distinct kinds of surfaces or properties in one nanoparticle depending on its use. So these are dendrimer-based nanoparticles. So these are dendrimers with metal or organic core at the center of it. So this is just an explanation or a brief overview of what types of nanoparticles are available that you can play as per your requirement or hypothesis. are other forms also, like quantum dots are there, and then there are semiconductor nanoparticles where they are given unique optical properties. So now I will speak briefly on ICMR's role in promoting research using nanotechnology. So as you can see on the slide, between the year 2018 and 22, we have funded over 250 proposals, research proposals, and they have come from various schemes. That includes the SRF or the RA scheme, where we have PhD or post-PhD students. Apart from PI, the investigator-initiated proposals such as ad hoc proposal, and we also had a task force which had different themes like theranostic, diagnostics, drug delivery. So those proposals, so there are approximately 250 proposals that have been funded during the last 5-year tenure by ICMR. The major focus is on what we did a review of all the data that we could compile from these funded projects, and what we found out that their major focus is cancer therapy, diagnostics, and drug delivery. So we also support clinical trials which are nanomedicine based treatments or interventions, and then we have also worked on development of ethical guidelines and safety protocols. That is again one important issue, the ethical point of view of using, why use nanoparticles if we can do with the normal formulation. So the ethical guidelines for using nanotechnology in medicine, and apart from the safety protocols. And there is a growth in patent filing, and ICMR is also involved in helping researchers across India for filing patents. So growth in patent filings also highlights India's growing contribution towards nanomedicine globally. So I don't know if this is visible at the end or not. So this is the hotspot map, which is basically highlighting the region-based distribution of the research that is applied from and funded from ICMR in the field of nanotechnology. So the darkest side shows that the maximum number of projects came from the pharmacy colleges from southernmost part of India. So this also shows that there has to be contribution that needs to increase from other parts of India, especially the northern parts, where you can really see the portion is very light in comparison to our southern applicants. So we did a review, and based on that scoping, we were able to identify the research gaps. The research gaps from this, from the distribution. This is just data that is based on those 250 funded projects in the field of nanomedicine. And what we found out that most of the projects are working on only meaningless research in the field of cancer. Most of those research won't see the light of the day when we talk about translational research. So this shows that people are taking up nanotechnology because it is an in area, that it is going to be promoted commercially in the field, but without having a proper in-depth knowledge of the field. People are taking up nanotechnology, and based on whatever research paper they go through, they just want to prepare a document, a research proposal using nanotechnology without any prior expertise in that, and hence that is something not going in their favor, because research should not stop at M.Pharm thesis or a PhD thesis. It should go to the next person who can take it up to the level where it becomes a product in the market. So the activities was again without focused drug delivery, non-quantifiable cancer research. So these were the major issues in the project. Most of them were either drug delivery or cancer research, without any hope for the next stage. Out of these only, there are 4 proposals which have gone into patenting or development into the next stage. The most important thing is when you prepare your hypothesis, it is very important you cannot be biased in what results you will obtain, but your hypothesis based on that review of literature and extensive scoping will tell you that whether it is needed or not, whether you are working in a health condition that needs priority, and whether your nanoparticle research is at all required in the field. It doesn't have to be something that you want to work in nanomedicine and you just pick up a health condition. Whether it is feasible or not, that is also very important. It will be taken up by industry or not, it is also very important because this is pharmacology. So, out of these 250, when we went ahead, what translational contribution came out of it, and there were nanocarrier system and biosensor research that demonstrated commercialization potential. Nanoparticle drug carrier research was there, and they proved that there was some drug delivery which was improved and reduced toxicity from that using that particular system. Then nanorobotic system studies, they could give a proof of concept for precision targeting of the disease cells. They were development of both nanoparticle sensors, so which help in when it's a proof of concept, that is they are taking up further. They have patented also. So early detection of microbial infections. There are startups and incubator programs. What we feel, startups and incubator programs can help us in bridging the gap between the research lab and the next stage of development. And ICMR is also encouraging partnership to overcome the GMP or manufacturing challenges when we come to nanosciences. So this is a big slide which is showing the challenges. So I will briefly take you through it. So there is what challenges we understand from this exercise. There is a fundamental knowledge gap. There is a fundamental knowledge gap in understanding of nanosciences. So the knowledge gap is there. You might read it as a theory, but what when you are going to formulate a nanoparticle, so the stability, the toxicity, the scalability, reproducibility, all these factors should be there in your mind. You might make it one time by looking at the literature, that is fine. But how stable it is going to be, can somebody else reproduce it, can you escalate the production? So all these things, do you have the capacity or do you have a collaborator to do it using good manufacturing practices? So that is one issue where there is a gap in the knowledge base regarding the nanosciences and nanoparticle interaction among themselves and their behavior when they will be in a biological system. So it is just not a petri plate. It has to be when you are using it pre-clinically and finally with the hope of using it in human beings. So there has to be an expertise behind it when you start working in the field of nanomedicine. And as I have already said, there is a big issue when we talk about going out from your bench to outside, that can you grow it outside the lab in an amount which can be taken up for human use. Then the next problem is the safety and the toxicity. So there is a big issue, and there are a lot of contrasting views on the toxicity of these nanoparticles due to their surface, due to their physiochemical properties. So that most of the studies, they end up just doing MTTSA. So we need more. So you have to go through a panel of toxicity studies to show that your product or your formulation is safe for use in not only in animals, for further research as well. And the biocompatibility. As I said, you cannot just introduce anything, reduce in a nanoscale, thinking that it can be given to anyone. So you have to prove the biocompatibility as well. Then we have spoken about human use. What about the environment? So the environmental impact and fate of the nanoparticles when they are excreted or the environment is exposed to these. So that is also one area one must look into when they talk about doing research or taking it to human use. Then there might be occupational safety and exposure issues when we talk about such research. So all these points should be taken care of because the committees or the experts who are looking into such projects or proposals, they tend to give you a lesser score if you have not taken care of to answer such questions. Then there are translational and applied gaps. It means whether your product can have clinical application. Clinical application, when I say, I mean whether they can have application in diagnostics, whether they can have their application in therapeutics or as a medical device. Then manufacturing, again the scalability. Can it grow in that particular form as you have proven to be safe and efficacious through a GMP facility? And obviously, because this is required for commercialization. Unless you commercialize your product, it will have no use. That is why we need regulatory and ethical guidelines in place, because you have to follow the standardization of such products and molecules under stringent guidelines and regulatory rules. So there have to be standard protocols. If a lab has 10 students and 5 of them are involved in the nano research, they should be following that particular standard protocol. You cannot have your way, so there has to be an ethics committee or ethics guidelines in place for doing the same. So we need to strengthen our regulatory framework as well. Then there are interdisciplinary and educational gaps. So we need collaboration. Collaboration means you, as a pharma student, you might want to do research, but there is physics involved, there is mathematics involved, there is pure chemistry involved. So there should be collaboration from these expertise when you want to get into nanotechnology research, especially in the healthcare. Then there is a need for developing an educational curriculum when we talk about nanosciences. There can be a nano research institute, but before getting into that research, one should have a subject on nanosciences to understand what it is, what impact it can have, and what you are doing has ethical and regulatory approvals. So, and there has to be public awareness and engagement when you get to the level of public health. When you want to introduce this particular molecule of yours after every guideline and regulatory framework it has gone through, then it should have that public awareness and acceptance of this particular technology. So nanotechnology is something going from a big TV to a mobile. So that microchip that your phone has, the SIM card that your phone has. So these all are components of nanotechnology. So just imagine if you want to do the same to human body, if you want to manipulate human body, you have to be much more careful. So as I said, regulatory efforts for development of nanomedicine is very well required, and ICMR has been part of 2019. There is a guideline which is freely available on the internet, so you can Google it out. This is nanopharmaceutical guidelines. It provides a framework for evaluating safety and efficacy of any research molecule. Then we need harmonization with global standards. So harmonization with global standards means FDA, Food and Drug Administration from USA, a European Medical Agency, or in India we have the Central Drug Safety Organization, which looks into regulatory approval. So we have to have a research, not just in animals on a petri plate. We have to work on something that can be used for humans and get approval under the various regulatory structures across India and globally. So there is a need for regular updates to match advancements in the technology. So what you are doing is not novel. It must be carried out across any part of the world where researchers are working on this particular subject area. So you just have to be in sync with the advancement, and plus the gaps. You need to fill in those gaps which we have already discussed. So we have spoken about the gaps and the challenges and what we can do. The next one is the emerging trends. So emerging trends can be, we are all talking about machine learning and AI. So artificial intelligence driven nanotechnology. Then there can be development of which is very well required with so much of pollution and the whole ecosystem imbalances. So we need eco-friendly nanomedicine. So biodegradable nanoparticles are required. Then in the top of the world is development of personalized medicine. So targeted therapy, especially based on genetic profiling, is very much required, and people are working on it. There is one example given at the bottom of the slide: a graphene-based sensor for real-time glucose monitoring in diabetic patients. So this is one kind of innovation which prevents or helps in non-invasive way of checking the blood sugar. So that is one boon for the diabetic person. So the future is we need to work on scalable technology. We need to bridge the gap between the stakeholders. We need to merge nanomedicine with the advancement in the field. We talk about diagnostics, whether therapeutics or medical devices. Then focus on long-term sustainability, definitely, and the ethical and safety consideration is a must from the day one when you want to focus on this technology. And apart from that, we have to preserve our future generations. So think this particular technology that you develop should be eco-friendly. That I would suggest. Next, please. So ICMR is at the forefront of promoting nanotechnology research in healthcare. There are efforts in funding, collaboration and policy development that have accelerated advancement in nanomedicine, but much more efforts are required, and there is a need, and we are continuously doing investments and are part of regulatory framework. This will ensure the safe and effective application of nanotechnology in the field of healthcare. So, I am done with the what is nanotechnology and what we are doing. It is just the last slide. In the research, the extramural grants are of interest to this organization as well. So, we find a major funding investigator initiative that fetches you around 2 crores. There's an intermediate grant that goes up to 8 crores for 4 years, and there's a possibility of establishment of Center for Advanced Research. This is for very senior people who have shown their credentials in a subject area. So you can include research very much from the discovery point of view to the delivery when we talk about, because we fund research in 4 wings, that is the discovery of it, the descriptive research, development and the delivery to the public use. So these are the 4, and there are different fellowships. Fellowships are for if you have a dental college also, the people who are doing MD, MS, MBBS, so there is medical fellowship funding. There are thesis writing funding for those students. Apart from that, we have funding available to conduct a workshop, not conference, but for workshop you are very much eligible to apply, where you are going to train people. So for training students and faculties, there is a possibility of getting that kind of money. And we fund to non-ICMR students for traveling to conferences. We have short-term 3 months to 6 months fellowship for non-ICMR researchers to go out and work internationally. So this is a broad overview of what we can fund. Next slide, please. So this is the pyramid of the grants that I spoke about extramurally. So that is very much available to even this college. And the deadline for small grant and intramural grant application is open till 1st of April. So all the researchers who are interested in applying for a research proposal, we will be glad that we can have application from this college as well. Next slide, please. The link is given at the bottom. The slide is here, so you can click on that. Next slide. So this is just the expansion of what I said. Next slide. Next one. Yeah. So securing your project funding, if you have to summarize, you have an idea, you do the proposal writing. All the advertisements, they come with a format of how your proposal should look like. So that you should frame your proposal. It is just not the idea how good it is also how good you are writing at it, plus your credentials, definitely. So you do not jump to any alien subject just because it is in the market. You should focus on what your expertise has been and what you have demonstrated previously. So you have essential elements of a successful grant proposal. You create a realistic budget. If you can do your work in 50 lakhs, why you have to put a 2 crore fund where a reviewer will eventually know that this work can be done in much less. So be realistic about your budget and you ensure that you adhere to research and clinical standards. Next, please. So this is the outline of the proposal, how your proposal should look like. Next one. So thank you so much. So this was it from my side.
Thank you, ma'am, for this wonderful talk. Now I request to our chairperson, Dr. Akhilesh Singh and Dr. Pooja for further proceedings. Please.
Thank you, Dr. Nehan. I request all the audience, please give a huge round of applause to Dr. Tabassum. It's a big... so much, ma'am. The topic of Dr. Tabassum's lecture was ICMR's role in promoting research in nanomedicine. Ma'am very well explained how nanotechnology plays role in revolutionizing diagnostics, drug delivery, regenerative medicines, etc. It was really very well explained. Ma'am also highlighted on the role of nanotechnology in medical devices and specifically medical industry. With respect to medical nanogenerators, medical devices, gene therapies, cosmetic industry, etc. She also explained on different forms of nanosystems, for example, carbon nanomaterials, quantum dots, fullerenes, graphenes, dendrimers, etc. Ma'am highlighted about the key initiatives and research programs. Over 250 projects are funded by ICMR during 2018 to 22 tenure. Major focus on cancer therapy, diagnostics and drug delivery have been covered under these projects. Most of the projects were funded for the southern state of part of India, and she has also impressed upon how being in the northern part, how we can write projects and fetch grants from ICMR. Then Dr. Tabassum impressed upon research gaps from distribution of ICMR funded nanomedicine projects across India. Due importance was given to challenges in nanotechnology. Ma'am also focused on the regulatory affairs for development of nanomedicines in India. Dr. Hina threw light upon the emerging trends, and she explained very well about the AI-driven nanotechnology, eco-friendly nanomedicines, nanotechnology, personalized medicine. Finally, future prospects were discussed. All the initiatives by ICMR were highlighted very well by Dr. Tabassum. Thank you so much, ma'am, for such a valuable, insightful lecture, and I am quite sure that we all have learned a lot from your lecture today. Thank you so much, ma'am.
Hello. Many congratulations, Dr. Hina, particularly for selecting the topic that the ICMR role in fostering the nanotechnology research. Because ICMR is the body which is concerned with the diseases and the treatment for the diseases, particular selection for nanotechnology that indicates that if the treatment is available in the traditional way, how it can be made better considering the bioavailability and the fast action of the drug. So that way the treatment will be more better for the different diseases. We have well covered the different areas, there were 14 areas, including the diagnostics, including cosmetic, dentistry and other areas, even the medical devices, how the nanotechnology may have contribution in the treatment of diseases in the better way. We have well covered the ethical guidelines which may be required for the research in the nanotechnology area and nanomedicines when these are selected for the diseases. The research gap you have well explained how it can be made, reduced, and the commercialization aspect also has been covered by you. The future prospects, including the regulatory efforts by the ICMR regarding the development of nanomedicine, which is the need of the hours. And lastly, you have given the tips for how the project funding can be done and how the proposals can be made for getting the funds from the ICMR. I again congratulate you and your contribution and selection of the topic for nanomedicines to give the better treatment of the diseases. Thank you very much.
Thank you, sir. Now I request our worthy director, Dr. G.D. Gupta, sir, and Principal ISFCP, Professor Dr. R.K. Naran, to please come on stage to felicitate our chairperson. First of all, I would like to invite Dr. Akhilesh Singh, Director and Dean, LNCT University, Bhopal. Please give him a huge round of applause. So, I would like to invite first our worthy resource person, Dr. Hina Tabassum ma'am, please come forward for felicitation. Please give a huge round of applause for Dr. Hina Kalasthi, ICMR Scientist B. Thank you, ma'am. Next, I would like to felicitate Dr. Pooja Chawla. Thank you, ma'am. Next, I would like to call Dr. Akhilesh Singh, sir. Thank you, sir. Thank you so much. Thank you, Dr. Hina, Dr. Akhilesh, and Dr. Pooja. So, let's move to the next session, that is Technical Plenary Session 7. For this session, I would like to invite our chairperson, Professor Dr. Pawan Kishan Sir, Punjab University, Patiala. Sir, please come on stage and please give him a huge round of applause. Our second chairperson of next Thank you, sir. Thank you so much. Hello. Thank you, sir. I request the chairperson, sir, please take your distinguished seats. For further proceeding, I would like to invite Scientific Committee in charge, Dr. Sankumar Verma.
Thank you, sir. I would like to introduce the speaker of the second session, Mr. C.S. Mujibbuddin. Mr. C.S.
Yeah? Is it okay? Is it fine? Is it a good morning or good afternoon? Don't tell good evening. We have some more time to tell good evening. But thank you. Good morning, everyone. So I am Moga, and I come from Hyderabad. So this is the first time I am coming to Punjab. It's all efforts by your team to cover. And give a round of applause for the discussion. Very overwhelmed with a lot of students engaging sessions and all. So I will not bore you. My time starts exactly 40 minutes. After 41 minutes, you can say that leave the room. I will leave. Don't worry. So exactly I will mark my time for 40 minutes. So let's see I can engage you for 40 minutes or not. And I will make sure that how your future for healthcare systems or pharmaceutical industry looks like. So most of you guys who are sitting in this room does not want to become pharmacists. You accept it or not. So everyone wanted to become either doctor or something like BDS or something like that. But might be because from childhood no one wanted to become a pharmacist. So once we are born, we were not being said that you are a pharmacist. Either if you see in a 3-8 movie, you will say that ladka hua to, ladki hui to. Now also we don't tell that pharmacist hoga. Hamara ladka hoga pharmacist, hamari ladki hogi pharmacist. The situation has got changed over the years, and we should feel proud that we are pharmacists. We delve into the research of discovery of either medicines or device or cosmetics or vaccines or nutraceuticals. You name it, we have it. So that's the importance of pharmacists, role played in pharmacists. And we are proud to be a pharmacist because I go to a lot of sessions. I speak to doctors also, and I give sessions to doctors also. I speak to engineers, I give the sessions to engineers also because they both does not understand the science behind the discovery and how the research is forwarded. So my topic today is very interesting. Before I start my session, I want to just tell one small story which happened in US. Everyone is interested to listen story, yes or no? Yes. Okay, I think ISF college are very excited to listen to the session. So there was 2 people in a room and both people got murdered and room was closed. And suddenly FBI came into the room and they saw that both people were murdered and room is closed from inside. And there is no third person coming inside or going outside. And FBI people found that there was one Alexa which was there on the table. So FBI got doubt that there was no third person, but somehow both people died. How? So they wanted to check with Alexa to know that have Alexa heard anything. Because the murder has happened 12 hours back, Alexa might have heard the murder voice. That's the doubt. Then these people went to Alexa and said that have you heard anything? Then it said no, we have not heard anything. Then FBI people went into the back end of a patent programming that how Alexa really works. Because Alexa has to work. It's a, if you ask some question, it gives answers. So then they went into the back end of a programming and they saw that Alexa listens everything, or it does not give answers. So if you go and say, Siri, listen something, it will listen. Sing a song, it will start singing a song. So then they found that there was one program which Alexa listens to your voice. And finally, FDA went to Alexa and said that you have your programming that you listen to voices. Now you tell me who are the people who, apart from these 2 people, were there in this room. Then finally, they have identified that there was one more person whose voice was being recorded in Alexa. And finally, the person who was murdered was being caught. So why I said the story? What was the meaning of the story? Why I said Alexa? Why I said Siri? So anyone use a Siri? Siri is beautiful or Alexa is beautiful? Both, we don't know they are beautiful or not, but both are females. I don't understand why the bots are not prepared by male voice. Always male voice is more disturbing than female voice. You find out any telecallers who are males. If you get any call from a male, you say immediately cut. At least a girl voice, at least you say, okay. You tell him your voice is so sweet. He explains your bank statement, everything. I might think about it. So that's the beauty of a voice which can be given. So why I said this whole story is because the future is all about chatbots. I'll tell you one more interesting story. Okay. So now 2023. during 2023. So ChatGPT. Everyone uses ChatGPT? So I think the best thing is to confuse ChatGPT. We are asking very difficult questions so that it fails in exams. So we are testing the ChatGPT, and in 2023, February 14. I think everyone knows what is February 14. Yes or no? I know you guys know about it. So, but the funniest story about ChatGPT is in 2023, 80% of love letters which were being written to boys to girls and girls to boys were not being written by them, but written by. And we are idiots thinking that it was being written poetically. So that was an interesting story about what? ChatGPT. And also one more I have said, which is called as Alexa. Now you ask me question, sir, why is this all stories? We are sitting in a conference which is pharmaceutical research, and we are telling about all chatbots, the audios, this ChatGPT and all those things. Because the future is all about how you can take care of a patient with the existing technology and how you can grow your technology. Remember one thing, you are sitting in this room. Tomorrow you are sitting in this room, and tomorrow you might come here and stand on the stage also. So to get into that position, first thing you have to know is first your technical knowledge, your pharmaceutical knowledge, as much as deep you can go for it. The second thing which we are applying, biggest fear we have is technology. Adapt to technology. Take a technology. See one beautiful thing. Everyone in this room are using Instagram. More than 1 or 2 users also. So one boy is having only one name, but he is having 2 more users with a girl name, so that he can blackmail other boys. Okay, but have you thought that any time we got one user manual how to use Instagram, how to send messages, how to share reels, how to use technology to upgrade ourselves? But no. That's the beauty of Indians. That's the beauty of youngsters. You can learn any kind of technology. With the pharmaceutical knowledge, you should have one of the beautiful things, that is what we call as technology. And with these 2 combinations, there is one more thing which is very interesting story. You know what? Future is about AI, but one thing AI cannot take from us. Any guesses what thing cannot be taken from AI? Oh, sorry. AI cannot take one of the most interesting things, that is what you call it as sales. Okay, so in an older stories, you should be best seller. Then you can get everything. But AI cannot sell you any product. AI cannot sell you any services. You name it, you cannot sell. You can't develop a business with AI. You can't say that, okay, system, you call everyone and then get the business. You can't do it. So now the most important third thing is presentation skills. Either it is the way you talk, the way you present, the way you project, everything matters, the third one. And the last one, fourth one, very interesting is the regulatory guidelines or regulations. Okay, either it is USFDA, MHRA, ESFCP, or DCGA, or ICMR. Guidelines, what are being done? Because I might speak very good about chatbots, but we are bounded to one thing which is called as regulations. We cannot go and say that, see, there is one person who can say anything. Okay, one person early morning 4 o'clock he will wake up and he will say that Newton's third law is wrong. Okay, you know who is that person? Any guesses who is that person? Newton's third law is wrong. So what is Newton's third law? Okay, so he can wake up early morning 3 o'clock and he will say that this is wrong in the whole world, and he is a famous person. Who is that person? Elon Musk. He might come and say that boss, Newton is also a human being. He might have forgotten to write something extra line. Why can't we test it now? That's the beauty of innovation, what he is doing. But we can't become Elon Musk and we can't say that no, I'll do whatever I want to do it. So we should be bound with some of the regulations. If you wanted to become the best person, the best version of you, the highest salaries, then you focus on this 4 things. One is pharmaceutical knowledge, the second one is technology knowledge, third one is your presentation and communication and convincing skills, sales and marketing skills, and last one is regulations. If you are best at 4, you can come any place. So this is the first time I am coming to Punjab. So why I came from Hyderabad to Punjab? Because of this 4 things. If you are delved into these four things, wherever you are, people might pick you and take you. That's the beauty of pharmacists. Be proud to be a pharmacist, then everything rest will come. Don't complain my college has not given, my institution has not given. What you have given to your institution. That's the beauty of what you have to have. I think I feel very proud to be here because when I was studying BCom and MCom, I was not having this kind of opportunity to speak industry experts. And my version was get more marks, more percentage, then I'll get job, or else I'll be jobless. I don't know what is happening in industry. That's the beauty that this kind of a conferences will come up with networking and you meet a lot of people, open your minds out of your books and academics and industry needs what are needed. So that's the beauty. Look at these conferences, the best place you are sitting, that's ISFCON. I am proud to be sitting here or standing here for NIR ranking 25 this year, sir. Let's give a round of applause for the best institution you are in. And you are in a station. So, why I spoke about these two stories, one is Chatbot and one more is Alexa. So, both are similar kind of things. What are the similarity in this? Is they are chatbots. They are not human beings, but they are chatbots. Everyone we are using chatbots, but what is the link between pharma, healthcare and chatbots? I am going to break it now during my session. So without any delay, my first session or my first slide is how chatbots have helped in healthcare system and also in particularly to pharma industry. See, I'll tell you a small story before I go ahead with my next slide. There is a patient who got one allergy on hand and he is running to a doctor, waiting for a doctor for 2 hours and then doctor came, has seen the allergy and then he wrote one lab report. He went into lab, given blood sample and then that report patient took to doctor and then doctor gave treatment, patient went home and he started taking a drug. Got my point? So now patient wasted 1 full day of time. Now I will tell you the story which might be your current or future. Now it is already happening. Future also. Now what happens? Patient got allergy. Patient took a photo, went into chatbot, held that. And then patient started saying my age is this, my gender is this, I am walking this, I am sleeping this, I am taking this medication, my allergy is this. Take my photo. Book an appointment, yes, to doctor. And chatbot is searching nearest doctor who is there and started booking and sent one message to patient saying that your appointment is booked. It's same message sent to doctor saying that your appointment for a patient is booked. Both got message. Immediately, sharp that time, both started opening the Zoom call and then patient started explaining that boss, I have a problem. So this is my allergy, you see it. Then doctor immediately said that okay boss, wait, don't worry. So he started writing all the signs and symptoms and then he started writing his lab test requirement. One lab test requirement sent to the nearest lab of the patient and the lab test requirement sent to the patient. Lab fellow who is nearest to the patient came to patient, he took the blood sample, he went back to lab. And then immediately the lab report was being transferred to a patient and also to a doctor. So doctor one more time started sending one appointment request, reminding saying that boss, your reports are ready. So then immediately both people came into Zoom call. Then doctor started saying that okay, your disease is this. You have identified what is your disease. And immediately after giving this disease, what happened? Prescription was being written. Electronic prescription was being sent to the patient. And now one report, one prescription was sent to pharmacist. Now pharmacist took a drone, put the pharmacy medications and then tapped the location of the patient. Drone went to the patient home and then patient took a scanner, started paying the payment and then immediately the drone opened, he took the medication, he started taking the medication. Now in this whole story, we have used multiple technologies. Starting from chatbots to blockchain technology to telemedicine to integrated services. All kinds of technology we have made so that patient travel is stopped and the accuracy is increased and immediate treatment can be given. So these are all things which can be done with your technology. So earlier story, we had so much of difficulty. Later story, we had, I hope we are all set. Yeah, thank you. So earlier story, we had this kind of environment where patient has to suffer. But now with the use of technology, now you have very good advantage. Now tell me, When a patient comes to you, what do? Families, how many people raise your hands? Okay, perfect. So now what do? You collect signs and symptoms and you start giving treatment. But now this is not story anymore. You cannot just say that, okay, you give signs and symptoms. Now try to understand patient is more knowledgeable than a pharmacist and doctor. But that knowledge is virtual knowledge because Google knowledge. They will read something on Google and they will come and say that my signs are this, I think my signs are cancer. How do you know that Google has said that? Or Chatbot has said that? So the point here is how can you crack this kind of a knowledge which is coming from the patient is more important. And patient to give a treatment, it's not just one word signs and symptoms. First, we have to understand his demography based on which gender, age group and the racism. Because see, Punjabi people and Hyderabadi people, there are a lot of difference. If you eat 3 times chicken biryani also, I'll have it. So, but here I have to eat kulcha. Yes or no? Or samosa, paratha, yeah, wonderful. Most interesting story is what food. Okay, so we don't find out boring when you speak about food. Now there is one thing which is called as racial difference. And apart from that, there is a second category which is already pre-existing diseases. What are the different diseases he is having? Apart from that, what are the medications he is taking? What are the food allergies and drug allergies he is having? And the third point, everyone is more of health specific and going and hitting the gyms and walking and running, all those things. What is physical activity? And what is the level of stress? It's not just his signs and symptoms. It's a story which you have to build into it. Answer this question, then you will get a response. What are the physical activities? How much stress level? How much is his screen time? What is his social habits? Does he smoke or drinks or takes any other things? Or how he works? Or what is the sleep hours? He sleeps before 12 o'clock, after 12 o'clock, how much junk food he is having? All those stories comes into picture. And finally, your signs and symptoms. With all existing data, you have to use a technology which is data science. With the existing patients who are already like this, we will get responses. That's the beauty that how your future is looking for pharmacists and how you have to plan for the treatment and also precision medicines. Because one shirt does not suit for everyone. How one medication is suited for everyone. It's about the precision medicine based on all categories, X, Y, Z categories. That's the beauty of AI and future of AI in your pharma and clinical research. So I'll focus only one word, which is called as chatbots. Can I have my slide? Okay, so introduction to chatbots. I hope everyone knows what is chatbots. All your ChatGPT is chatbots. But how we are going to implement this chatbots in pharma and clinical research is something which is an interesting story. So what is chatbots? AI-powered tools which can give answers like humans. It does not require a human, but it can give answers like human. So now say for example, patient is asking a question at midnight 12 o'clock. Some of the students call me at midnight 12 o'clock and they say that, sir, you have jobs or not. So now this is a big question to ask at midnight 12 o'clock. But if I have my cloned version of ChatGPT of Mujib, he can give any answer at any point in time, 24/7. You chat with me. Okay, but I am not Mujib, but a cloned version of chatbot which is Mujib. So that's the beauty that 24/7 one system can work. And one thing I can tell this session one time, 2 times, but not 100 times. How can we take this data and reproduce at every conference is something which you have to think. Why should we waste our time? I can develop one more session to next conference and how we can use this conference and develop on chatbots. That what Mujib has spoken and how we can put step by step. That's the beauty of chatbots and how bots can be developed. So I have spoken about ChatGPT and chatbots, why they matter in healthcare, because in healthcare, patient does not wait. Patient cannot wait for a doctor, patient cannot wait for a pharmacist. So initial preliminary screening and initial questions to a patient or a healthcare professional or to pharmaceutical companies can be answered by your chatbots. That's the beauty. how in this room can develop a chatbot, you should start thinking about it. One product, one tool, you can become biggest person to be paid in the industry. So that's the beauty. Okay, so I think, yeah. So now this is the different types of AIs and also chatbots. I'll not speak much about it. The top layer is super intelligent AI, which is more of theoretical. It just acts as theoretical one. The second one is the strong AI, which is human-like intelligence, which can think like human-like. And the third one is the narrow AI, which is weak AI, which is chatbots. The chatbots is simple example is chat. Okay, but how chatbots can be developed only for pharma? What is the best advantage? How we can use it is something which we are looking at. So now chatbots fall under the narrow AI. They use what language? So NLP is the technology which you have to understand. Okay, so in this classroom, at least if you hear about NLP word, you are successful. Because at this particular time, can you book an appointment? And you develop this kind of a chatbot which can automatically book an appointment without any delay. So chatbots can directly book an appointment based on Google reviews also. Because if a doctor is giving more of Google reviews, then he is the best doctor. You can pick it up and you can book an appointment. Automatically system can book an appointment, which is chatbot. So that's the second one, automated booking and reminders to patient improve the attendance rate. And one more thing, chatbot can give reminders. Saying that boss, your appointment is 1 hour ago. After 1 hour, you have to read this. If you book a cab, you can do it. So now you can do one more thing, very interesting thing. If you have automatically booked an appointment, you can automate this with Uber or Ola also. Saying that boss, you are 1 hour away from this appointment. Your doctor stays nearly 4 kilometers. You have already sent a message or reminder to a doctor. He is ready. And if you want, you can book an Uber cab also for you. So if you say just book an Uber, it will automatically book an Uber. You don't need to put everything on calendar. Everything is booked by your chatbot. It can be done. And this is being done in multiple conditions. So that's the second one. And the third one, most biggest problem. In this room, everyone is sitting and smiles. But the biggest problem after 9 o'clock or 10 o'clock or 11 o'clock, where we are alone, we are struggling with one of the biggest things. That biggest thing is mental health. No one is talking about it. And everyone is talking about all big things in conference calls and how we can grow, how we can get crores of salary and everything. But the most important and basic need, which everyone is struggling from inside, is mental health. I might smile in conference, but what I do after 10 o'clock or 11 o'clock, no one knows. So how can I make everyone to come into a mode where everyone is challenged and having an objective? Mental health, we can develop chatbots, because mental health is so dangerous that we don't go and tell to our friends also. But you can speak with chatbots. So say for example, a person has searched a word called as how to do suicide, for example, in Google. And then immediately I can run a programming along with Google saying that if this word is being searched, immediately show an ad that you need a mental health support chatbot. Then they will click on it. Then chatbot will start speaking to them, saying that what is your problem? Tell me your problem, what happened? Why do you want to think about negativity? What happened? You tell, I'll give answers. It makes engaged, and there is no human being. It does not leak your secrets. So human being leak your secret, but chatbot will never leak your secret. You can engage with chatbots. That's the beauty, that you can start taking help from mental health programs also. Yeah, so now one of the most interesting things, how it can be used in pharma. Okay? So the first and foremost thing on left hand side, drug information. So now say for example patient called, suddenly patient called from US, saying that boss, I took atorvastatin, but I should take only 150 mg, but I took 400 mg. I should take this food, but I have taken with alcohol. So I want to know what happened. Now there is a pharmacist who is sitting and who is doing a research on this molecule which is atorvastatin. Patient called directly, medical information call center, saying that what happened. Then pharmacist is running for information. No need. They can develop a chatbot which with one word saying that I want to know all details about atorvastatin. Can you give in combination what happens to be taken? Then this pharmacist can immediately respond to the patient. It's not gender bias. I am not saying gender bias. It's always, see, the future is everyone in India also wants nurses from Greece, okay, or from a country like Israel, because the way they look, the way they get convinced is more good. So that's the beauty. So, but now hospital, I cannot have one receptionist from Russia or Iran or Israel. I cannot have that person because I don't have money. So, but I can have a chatbot there. Okay, so a patient can come, there is a machine there, automated machine, and then patient can say that, boss, I want to meet this person. So my details are this, my ID is this, my EHR are this, my EMRs are this, my scans are this, my follow-up is this. Can you book an appointment? Then it will start saying that, wait, I am talking to your doctor. Immediately chatbot went to doctor, sent a WhatsApp, saying that, boss, are you ready for one more next appointment? Then a doctor will say that, no, you take 1 hour time. The same message is being given to patient. So now this is the beauty, what you can do for the future. Hospital industry also we can use. The third thing, patients can use. Because patient can directly book an appointment, talk to the signs and symptoms and get the best doctors and report an advertisement, take a mental health support, different kind of things can be developed with chatbot. By sitting here, all the drug advertisements you can develop on chatbot. Anyone who thinks like that, everyone knows that we need new drugs, new molecules, either it's medications, devices, vaccines, cosmetics, or new cosmetics. But to get this, we need to do a preclinical research and also clinical trials, and we need to have the patient. Getting the patient is the most difficult part, because patient is sitting somewhere and we don't know. So recruiting a patient with chatbots is so much easy. Patient can go to a chatbot and say that my age is this, my gender is this, my disease is this, my conditions are this, I am taking this medications and everything. Chatbot can take the data and give it to the clinical trial people and include those patients into clinical trials. Within this, that's the beauty. You don't require big data to be collected on paper saying that this person is eligible or not eligible. We can use in clinical trial recruitment also for the patient. And the last one is the professional support which can be given for the healthcare professionals. So 10 minutes more. Yeah. I can take another 15 minutes, right? Yes, sir. Oh, thank you. So I will take 15 minutes, guys. So then one more is professional support which can assist the healthcare professionals with all product information. Like for example, I don't need to by heart everything. See, I feel like I don't remember anything from my B.Com or M.Com what I studied. I only understand how to apply knowledge, how to develop an innovation product, and how to convince people for my product. That's what I understand. So wherever I go, I prepare myself in this regard. The next slide. Yeah, and here one of the example is Pfizer is having already chatbot which is being developed. Pfizer has already developed one chatbot which can be given to the patient. That's the beauty, that how data collection can be done, analyze the data and given it. See, your future jobs which are safest jobs, are associated with only one word. That one word is data. If you are associated with a word called as data, then your jobs are so good. Either you are clinical research scientist or data scientist or data analytics or data visualization or data predictions or data analysis, your jobs are safe because data is huge. All data which is collected from hospitals can be analyzed and predicted. All data which can be used and predicted. That's the beauty. The next one, ongoing engagement. See, patients, why they leave doctors? Because doctors does not talk nicely. Doctors does not give time nicely. See, if they engage, if they make sure that patient is happy, then next time he will wait for the doctor and talk. That's the engagement you should have. Highest engagement can be done with chatbots. Every time doctor cannot call him and say that, have your coffee or not. So the chatbot can start conversation with the patient saying that you had coffee, you have your appointment with the doctor, you have to take this drug, if you have advisement, report to this, if you have this problem, take this action. This can be done with the patients with the chatbots. That's the beauty that how the future is looking. Next slide. Yeah. So how chatbots work? Very theoretically, how chatbot works, whenever you give question to chatbot. See, remember the future is about prompts. Okay? Everything is available in ChatGPT, but what question you are asking should be understood by ChatGPT, then only it gives accurate answer. Try to understand. Okay? And chatbots can give you words, text, Excel sheets, images, sounds also. That's the beauty. But we are just using one thing, text to text. It can give sounds also. It can give images also. It can give Excel sheets also. So now say for example, I want to know ISF placements from 1980 till now in ChatGPT. If you ask this question, it will go to the website and also develop it in an Excel sheet and give in 1 minute or 10 seconds. That's the beauty. Use it to the utmost of chatbots. You will get that answers. That's the beauty. So how we can use it? You put a query, you put a question, and the second one, NLP goes and processes it. And the third one, you get a response, and the last one, user receives the answer. So this is the one thing. And chatbots, every time it breaks answers. Okay, first what is ISF? Okay, first it takes ISF word as common, and what? It takes two questions, and then it goes and searches in the website and gives answers. That's how chatbots work. Think logically. Innovation is the best thing you can give now. It's not future about how you are going and getting these things. It's happening now. So this is how your chatbots work. Next slide. Yeah. So what are the advantage of chatbots? I'll have one short video here. Before I go ahead, can we play this video? It's a chatbot which is already being developed. You will be surprised to see all these answers. Yeah. Can we have that image, please? Browse YouTube. Yeah. Yeah. It's made very clear that how is future looking at it. So the future is doctors, hospitals, patients can develop their chatbots. That's the beauty. And remember, if not we, someone else. You cannot say that no, will not develop, everyone's job will be lost. No jobs are lost, it's more created with technical knowledge. We can invest our time in research, developing new conditions, because the way the human diseases are growing and the way the scientific evidences are growing, it's huge. Now it's not just saying that okay, one symptom. Patient is about polypharmacy. He's not taking one drug. He's taking hundreds of drugs and he's having hundreds of diseases and he's having hundreds of things to be reduced. That's the beauty of polypharmacy and the role of pharmacists to be played here. Okay. So now I have already said that what is the advantage for the patient is this. Healthcare providers and pharma research also. Next slide. Next slide. Yeah. So I think you can see the left side last image. I think right side of last image. So can you identify anyone? Who is that person? Right hand side last image. Okay. So it's me who is trying AR and VR. So which is augmented reality for the patient care. Now whatever I spoke is not future, it is happening now. So this photo was being picked I think 1 week back, which is virtual health assistants. So what happened here is they gave me a glasses, I wore the glasses, and then 2 points. The first point is it goes in a way that patient will wear these glasses, he comes to a place which is a front office or reception. And he can ask any question. And there will be a person who is a virtual assistant. It will be really like a human, beautiful lady who will be speaking for you directly. Then you can walk. By wearing the glasses itself, you can walk. You don't need anything. And the second thing, this is front office. The second thing, what happened here is I wanted to test taking a needle and putting it into the patient. That's my test to be done. But earlier how we used to do, we used to take a needle and put it into the patient. If he dies, okay. If he's not dying, let's leave. But now it's not true. You have augmented reality glasses where there will be a patient on bed and there will be a tray of glasses. I mean there will be injection needles. You wear the glasses and you have the button. So the one which is in my hand is the button. I can move my button to the injection needle. I can pick that. I can come back to my patient, start giving injection. It is called as digital twin. You can learn with digital twin and digital technology without putting into a direct patient by reducing the pain and giving more simulations. That's the beauty and it is happening now. So that's the virtual health assistance. Second one, you can improve your learning with virtual health assistants. It does not require really a patient or other models. Simulation model can be done. And the next one, surgical assistance can be done. You can use your glasses and do a surgery. And your chatbots will start explaining. Augmented reality will start explaining. that you have to cut this weight, you have to do this kind of things. It will support you. That's the beauty of the future. Next slide. Yeah. So now, how chatbots are improving the actions? The first point is it can give initial patient contact or personal contact. You don't need a receptionist. You don't need a receptionist going forward. You don't need a nurse. So first point of contact. Second one, it can assess the symptoms also. You don't need to waste your time to a pharmacist or doctor for assessing the symptoms. It can take the symptoms. The third one, it can give some care recommendation also. Try to sleep for this or do this exercise, yoga and all. And the fourth one, it can give follow-ups also. It can give reminders and follow-ups and everything. Next slide. Yeah, so machine learning, what are the biggest advantages of AI, ML and chatbot? The first one, it gives training. Second one, NLP process, user interaction, feedback and improvement. Next slide. Yeah, so this is an interesting slide because I spoke very nicely about future, that, this, but the biggest problem is, what are the disadvantages? What are the problems? What are the challenges? The first and foremost challenge is data privacy. We really have to be sure about the patient data because the amount of data which we are collecting is broken, then our lives are on main road. So because we don't know who is having what diseases. That's the data privacy and security. You should have a very high-end security system. Future is about data security and privacy. That's the importance. And the second one, information accuracy. It might be sometimes give a wrong values. It might be give a wrong prediction. And the third one, bias. There might be some bias which cannot be prevented. And the fourth one, adoption resistance. How many of us, our parents, are ready to adopt it? Chatbots. We might not be ready because the way internet has grown into Moga is not up to the villages. It's not about tribal places where this internet or mobile phones are being used. And how the education system has to cross the barriers have to be understood. So one of the most important difficulties is technology adoption. Youngsters are adopting, we are adopting, how we can adopt it. Next slide. Next slide. Yeah, so future, what is the future directions? What might be the future of chatbots? The first one, we can have voice-enabled assistants like Alexa and Siri. The second one, wearable integrations. You can have watches which can directly get integrated with chatbots. So your watch data is directly integrated with chatbots and give the answers to the patients or to the doctors. And the third one, predictive analytics. You can do a predictive analytics with all data collected with it and answers and personalized medicine also. Next slide. Next slide. Yeah, so these are the references which I have taken and you can take the slides. I will be posting the slides in Universal Research, my official LinkedIn, and you can contact me on LinkedIn. I'm available on LinkedIn and Instagram. Use more of technology to be contacted. My name is Mujibuddin Sheikh in LinkedIn and as well as in Instagram. You can follow me. You can ask me any questions in LinkedIn. If possible, if you wanted to get connected in Instagram, because I might not be very active. Instagram is only for Gen Z and I'm more of the previous generation person. So you can contact me in LinkedIn and please do ask questions. If you have any questions, you can ask me. I'm open for questions. And these are the references. I'll be posting the slides in my LinkedIn account and you can download from there. Next slide. Next slide. Yeah, most interesting slide and everyone is ready to clap. Now we can do it. So thank you so much.
Thank you, sir. Thank you, sir. Now I request to our chairperson, Dr. Felicitation sir and Dr. Arun Gora sir for their concluding remarks.
Give a big round of applause for CS Dignitary sir. He has explained very well in that part. But I have concluded
I also request our chairperson to please join. Thank you, sir. Thank you so much. Now I request Director Sir and Principal Dr. R. K. Naran to felicitate Dr. Pawan Kishan, Professor, Punjab University, Punjab. Sir, please. Thank you, sir. Next, I request Director Sir and Principal Sir to please felicitate Dr. Arun Vora, Director, Baba Philips College of Pharmacy, Ludhiana.
सर थोड़ा ये मूव करें इधर नु हाँ जी सामने जी
Thank you, sir. So, move to the next session. I would like to invite So we have our worthy respected chairman, sir, Shri Praveen Ji among us. So please give me a huge round of applause. So move to the next section. I would like to invite our next renowned scientist and our next resource person, Professor Saranjit Singh, ex-professor and head pharmaceutical analysis, Naipur Mohali. Sir, please welcome on stage for release the MOU. I request Professor Saranjit sir, please come on stage. I also request our worthy chairman, Shri Praveen Ji, sir, please come on stage for release the MOU. So I would like to invite Chairman Sir, Director Sir and Principal Sir to please felicitate the renowned scientist Professor Saranjit Singh Sir. Please give him a huge round of applause. Now it's time to release the MOU. The first MOU is in between and it's a proud moment for all of us. State Government University, Baba Farid University of Health Science. So please give a huge round of applause. From Baba Farid University of Science, we have Dr. Vinay Chawla, Principal, and we have Dr. Pooja Chawla, Professor from Baba Farid University of Health Science. Please, ma'am and sir.
हाँ जी सर
Thank you, Dr. Vinay Chawla sir and Dr. Pooja ma'am. Next MOU is in between Maharishi Manikeshwar University, MMU Moga. From there, I would like to invite Professor Vipin Stanley sir from MMU. Sir, please welcome you on stage for releasing the MOU between MMU and ISFCON College of Pharmacy. Please give a huge round of applause.
थोड़ा आगे थैंक यू
We are very thankful to Professor Dr. Vipin Saini sir. The next MOU is in between Guru Kashi University. From them, I would like to invite Dr. Manoj Kumar. Please come on stage to release the MOU between Guru Kashi University and ISF College of Pharmacy, Moga. We are very thankful to Dr. Manoj Kumar and Guru Kashi University. The next MOU is in between Clinosol Research Private Limited. From them, I would like to invite founder and CEO CS on stage. Please give a huge round of applause. I would also like to invite Dr. Saurabh Kose to please come on stage to release the MOU. Thank you, Dr. Saurabh Kose, Clinosol founder CEO CS2015 sir. Thank you so much. Next MOU is in between Sandeep Foundation, Sandeep Institute of Pharmaceutical Sciences, Nasik. From them, I would like to invite Dr. Manish on stage on behalf of Sandeep Institute of Pharmaceutical Sciences. Sir, please. Next is also internal coordinator Dr. Manish in between ISFCP University, Moradabad, and also proceed by Dr. So we have Dr. Kuldeep from Sandeep Foundation. Next MOU is in between Matish Healthcare Baroda. For this, I would like to invite internal coordinator Dr. Manish to release the MOU. Next, I would also like to invite Dr. Manish as an internal coordinator to MOU between ISFCP University in Moradabad and ISF College of Pharmacy. Next MOU is in between NRI Institute of Research and Technology, Bhopal. Internal coordinator, I would like to invite Dr. Vivek Asate, and in between ISF College of Pharmacy, Moga. Sir, please. Next MOU, this is the last MOU for release. Signa Pharma Private Limited. I would like to invite Dr. Priti Patel to release the MOU. Thank you. Thank you so much, respected dignitaries. I would like to invite all dignitaries, principal, deans, HODs are requested to please come on stage to release the. I request to all principals, deans, HODs, directors, sir, please come on stage to release the International Cyclodestinate 22nd Conference on 20 to 26 October 2026. Sir, please. I request for faculty members, please. I also request to all faculty members, please come on stage to release ICMR. Now it's time for first announcement, International Cyclodextrin Symposium to be held at ISF College of Pharmacy on day 27. Please give a huge round of applause for this first announcement, ICS 2025.
जाइए अगे नु
that we can organize this event at a grand level. Thank you very much.
Thank you, Director sir. Now I would like to invite our chairperson of this technical session. For this, I would like to invite Professor Dr. Anil Kumar sir, University Institute of Pharmaceutical Sciences, Chandigarh. Sir, please welcome. Next chairperson, I would like to invite Professor Dr. Vipin Saini sir from MFU Ambala. Sir, please welcome on stage. I would like to request our worthy director and principal Dr. R.K. Narayan to please thorough welcome to our chairperson. Sir, please. I would like to invite Dr. Sam, Scientific Coordinator, please proceed the session.
Thank you very much, sir. Firstly, I would like to introduce Professor Dr. Saranjit sir as a speaker of this session. Dr. Saranjit sir is a renowned figure in pharmaceutical analysis, particularly known for his expertise in drug stability testing, degradation, chemistry, and impurity profiling. Sir has served as the acting director, dean, and head of the Department of Pharmaceutical Analysis at National Institute of Pharmaceutical Education and Research (SAS) Nagar from August 1994 to October 2021. Sir holds a PhD and received the University Gold Medal from Punjab University, Chandigarh. Sir has over 40 years of experience in teaching and research. Dr. Saranjit published approximately 230 research papers, articles, and book chapters, and holds one patent. His research has influenced Indian and international pharmacopoeia. Sir has guided 135 masters and 15 PhD students. Sir has served as a temporary advisor to the World Health Organization and a member of expert advisory panel on the international pharmacopoeia, widely traveled across the world for sharing his learning at various forums. Sir served as a contributory editor and reviewer of number of reputed journals, including Trades in Analytical Chemistry. Sir has served as a board member of studies of multiple Indian universities and institutes. Sir executed 100 sponsored projects from industry wide at NIFER and delivered 612 lectures globally. Sir is currently a recognized industry trainer and consultant. I heartily welcome you, sir. For this session, I also welcome Dr. Anil sir, Professor Punjab University Chandigarh as a chairperson, and Dr. Bipin Sari, Professor Mahasmarkandeswar University Ambala as a chairperson. So I would like to hand over the dais to Professor Dr. Saranjit sir. Sir, welcome sir.
I think you are all waiting for lunch and there is no response back except one. Very good afternoon. That is good. That's good. It was very interesting to listen to Mr. Mujib coming from Hyderabad, and he started his discussion with Alexa. I had a very interesting experience with Alexa, and that too happened in Hyderabad. My son was in Hyderabad at that moment, unmarried, and myself and my wife happened to visit him. And he was on the first floor, and we went over upstairs, and he opens a room and says, "Alexa, put on the lights." And then he says, "Alexa, put on the fans." And we entered over there, and we are searching for Alexa. I went to, because I said, "Who is Alexa? How come he's a bachelor?" And this lady who is responding, "Your lights are on, the fan is on." And then I went to check in the bathroom below the bed. Then he really introduced me to Alexa. Like, "Papa, this is a device I bought for 6,500 rupees, and that can take all the commands." Of course, there are chips in the fan and the light that you have to give command to Alexa to put on the light. So it was very interesting that this device, now it was a large one, but now it is very small one. So, and that was what we call an innovation. And Chatbot, which has been discussed also, is a very interesting innovation in health sector and with application in pharmaceutical sciences. Now, my two requests before I start my presentation: one, there should not be any cross. And preferably, please bear with me to sit in your seats and not move out till I complete. I will try. I have been given 1 hour, so be ready that you have to have lunch only after 1:30, not before that. Okay, this picture, just to have a relaxed environment inside. Actually, what I'm going to talk. Okay, all right. Now, what I have brought to discuss with you, and this title, Dr. Gigi Gupta has been after me to give him the title almost 15 days back. I said, "No, I cannot give the title. I will decide and then give you." And this title was decided at 11:50 p.m. yesterday night. So what I'm going to talk is the innovation landscape and whatever is written over here about fostering collaboration in pharmaceutical research to promote innovation. It's a slightly different, but my focus today is not on AI, but overall on the innovation. Now, first to talk of innovation landscape. It was very interesting to prepare this lecture to find out what kind of innovations are going on and what exactly is innovation. Sometimes we are not very clear about the definitions, and it is very interesting to know about the definitions even. Defining innovation, now this is very important for every individual who is present in the hall to know what is the real definition of innovation. The innovations definition, I don't know, because these are getting disturbed. Can you shift to PDF file? Just open the PDF file. I'm sorry, because when you transfer from Mac to normal laptop, this sometimes happens. Okay. It cannot be put into the slideshow time. Good. Leave it over here. Okay. Foremost, read every word of it. What is innovation? It is the practical implementation of ideas that result in the introduction of new goods or services or improvement in offering goods or services. Now, the most important are the two words. That is practical implementation. So you are introducing something new, and you have to make sure that it is practically implemented. Innovation is not innovation if it is not used in a larger way, in a broader way. It is not put to use. Now here, there is some relationship between which I want to highlight. What is idea, creativity, invention, innovation and discovery? What is the relationship between them? How many of you really understand this relationship? It's very important when we talk of innovation. So we have these terms very normally used. Idea, creativity, invention, innovation and discovery. Now there will be some learning when we go into the understanding of these terms. Now, look into this. This is a very, very important slide. It says idea. Idea is simply the belief. Yes, this can be something new. This can be taken up further. So, we sit together, develop an idea to solve a problem. Okay? And here we have a belief that it will work. Now, it can be a theory coming out of some research which you have done, related research, or the science. The idea you are taking from some science, that this can be a new phenomenon. So let us go for it and search out for it. Based on the idea and the maturity of idea in your mind that it is workable, we go into the creativity. And this we call as invention. Invention can be a design, it can be an engineered product or a prototype. Actually, it is a prototype. So you create something as a part of invention. You have an idea, that idea is workable or not. Now, a very interesting, one of my students after the class came, he was from the pharmaceutical technology side, he came to me. Sir, you are in analytical side. I have an idea. I said, what is that idea? I want to convert a male urinal pot into a testing lab. While you are urinating, your all urine tests are simultaneously being done. This was his idea, paper idea. I said, do you have a paper idea? Yes. So he showed me the male pot and all the sensors which can be connected to that male pot so that there can be the urine testing done. There are huge instances, number of instances when a person doesn't know that I am into CKD, chronic kidney failure. So once you are getting a regular, using a regular male pot and it tells you that the values are now dithering and you are going to get into a problem. So now he asked me, sir, how I to solve it? I said, you have to first go to the college, you have to find out what test and what are the reagents used. And then you will have a mechanism to throw the reagents into the where the urine collects, some passage has to be there which goes into intact the urine and inside the some type of test and result. So idea goes into building up a prototype and he will build up a prototype based on whatever the possibility is. That is invention. It is not innovation. We misnomer, as a misnomer we use it. Then we go to experience. Once this creativity has been done, prototype has been produced, you go to the 5 customer, 3 customer, your family members, let us use it. Now once you have the experience of it, then you start talking, and it's a successful experience, you start talking about commercialization, delivery and larger acceptance. Then only it is an innovation. Mind it, before it is widely used, it is not innovation. Whatever you do in the lab is not innovation. It is only invention. That is what. Next, if you go, what is the difference of these terms from the word discovery? Now you are always talking about drug discovery, drug discovery, drug discovery. Is it the right word? Drug discovery means it's a right word that we talk of discovery. Now, here, when I put it to in this, here I am talking of what we say idea, art, creative ideas, invention, innovation. In this 4 circles, discovery is put out of it. So discovery is placed out of the what I have discussed till this time about idea, invention, innovation, and discovery is something different. That's very important for everybody to understand. When we say drug discovery, what exactly is discovery? Okay. Defining discovery, now it's very important to understand the definition of discovery here. Discovery is uncovering something that already exists but was previously unknown. Believe me, discovery is something, you are discovering something which is pre-existing. You are not preparing a prototype, you are not preparing something as a workable solution. So discovery is uncovering something that already exists but was previously unknown. Discoveries are often made observation, exploration or research. For example, the discovery of gravity by Isaac Newton or the discovery of DNA structure by Watson and Crick. It was pre-existing. The gravity was pre-existing. But Newton observed from the falling apple that there is something, a pull, which Earth is pulling an apple downwards Earth. So that issue was very interesting, and the discovery is something which is already existing, and you are unraveling it. That is discovery. Now, when you say drug discovery, and you have synthesized something new, you have tried pharmacology, done the pharmacological testing, is it truly a discovery? Now let's look into that. Now here I am giving you examples of, yes, there has been important discoveries in medicine. Penicillin. Penicillin was discovered because it was a pre-existing. There were microbes on the petri plate, they were releasing something which was killing the bacteria on the plate, and it was pre-existing. It was a natural phenomenon that was happening, that it was being released by the microbe. Then we had insulin. It is a part of the body system. Similar is the case of ether. It's a morphine, steroids, they all were pre-existing. Nothing that you synthesize. Of course, steroids are now synthesized, but originally it was a discovery of something which was pre-known. It was not pre-known, but pre-existing and developed by nature. Let us say, I will give you some examples of the difference between discovery and innovation. First is FT Raman spectroscopy. We have just observed the National Science Day. Okay. Now, Dr. C.V. Raman, discovery of the scattering phenomena and Raman effect are the natural phenomena. The scattering effect, that when a light strikes a particle and the light scatters, is a natural phenomenon. Nobody discovered it. It's happened. Your sky is blue due to what? The sunlight is coming, it is scattering light, and what we observe as a blue light. So what, if you see over here, Everts and the discovery what has happened, all it started from 1871 when Rayleigh scattering first described. It was not discovered, it was described here. And then you see 1871 going further, Sir Dr. Raman awarded Nobel Prize when he in 1927 again Crompton awarded Nobel Prize in Physics. This way, 1928 was the time when C.V. Raman and K.C. Krishnan discovered the Raman effect experiment through Raman effect experiment on 20th February. So what happened? Natural phenomena was put under some system to observe that. So you can see there were 4 Nobel Prizes that have been awarded from 1927 onwards till 2009. What Dr. Raman did, he proved the presence of scattering, and what Raman effect was, he proved that the light, when it is of a single wavelength, like I have this wavelength, this here, when a very high energy light is pushed through an organic molecule, the molecule scatters the light, and a very small element scatters from a different wavelength than I have an incident wavelength. Red is something around 700-800 nanometer. So what happens, he saw that he had put this sunlight white, and when there was a violet filter, and this is scattering liquid, you get a violet measure of it, a very small fraction is removed from the incident wavelength. Now this was the whole Raman effect that he observed. Very small element of light shows a different wavelength. It means this is due to skeleton of the molecule. Your IR, infrared rays, are due to the functional groups. and this is due to the skeleton buildup. Now, both Raman and IR are complementary to each other to provide a structural information. Now, that we call as innovation of Raman spectrophotometers. What Dr. Raman was having a 4-room size machine, now they are miniaturized, and we have a very small size Raman spectrophotometer. This is 1988 Raman. This is first standalone translation Raman system in 2009. Then first handheld Raman spectrophotometer 2015, just your mobile size. And first high sensitivity qubit-based measuring benchtop Raman system, which was developed in 2017. So, this is one typical example for which we celebrate the National Science Day. Second example is a very current example of anti-diabetic and anti-obesity drug, which we call drug series, which we call as glucagon-like peptide 1 agonist. Now, these are the class of medications primarily used to manage type 2 diabetes and obesity. Now, if you look into the historical, the GLP receptor, which is inhibited by these inhibitors, drugs, there's a long history of that. GLP-1, and it goes to as early as 1859 when pancreas was the site of diabetes. And continued the research, discovery of secreting in the first hormone, a gut hormone may stimulate the endocrine pancreas, 1905. Evidence of an insulinotropic gut hormone, 1930. Like this, the GLP-1 was, as an incretin, was identified in 1973. This is discovery, a basic physiological phenomena which was identified as GLP-1 structure. Subsequently structure, there is something which is influencing our diabetes. Then comes the innovation of GLP-1 agonist. So, question comes over here is, we have discovered a physiological phenomena against which we target our drugs, we develop drugs to target those. So, the original innovator here of glucagon-like peptide GLP-1 agonist was the pharmaceutical company Amalin Pharmaceuticals, which developed the first GLP-1 agonist, exenatide, approved by the US FDA in 2005. Now, this company was purchased by AstraZeneca. Here again, the journey starts from somewhere 1965, 1980, then goes to 85, 90, and the first drug, exenatide, whose synthetic extending 4 as a diabetic therapeutic was invented. Now, here comes the GLP-1, liraglutide and semaglutide. You see over here, I just want to highlight to you. Here it is written in 2004, these are the scientists who are involved, for Novo Nordisk, semaglutide invented by Novo Nordisk scientists. What invention has been used? Up to that time, semaglutide was not made a broader drug applicable. It was invented, then it was put to the market for wider use, and then we will call innovation. And these two Novo Nordisk products are available in Indian market under the semaglutide tablets, Rybelsus, and liraglutide, that is Lirafit. So these two products are already available for managing diabetes as well as for anti-obesity. Now, we had a discovery of GLP receptor, GLP-1 receptor, we had an invention of the drug which would work against it, and then it was eventually the innovation that semaglutide would be put into the market. Now, it is a very interesting story. My wife, we went for a medical checkup, my wife was diagnosed to have an early glaucoma. And glaucoma is an incurable disease. Only you have to instill a drop to manage the eye pressure. Now, she's obese, a bit obese, and she was prescribed semaglutide for that reason. 6 months after she was on semaglutide 7 mg, we went over to the doctor back for eye glaucoma. She said, you come after 6 months, and when we went over there, she said she was surprised that your glaucoma is getting reversed, which is never heard of. So it was very interesting that glaucoma can be reversed. And then, as a pharmacist, I checked on Google which drugs to do it. And the only drug which is added is semaglutide. And found a paper where it was very interesting. A paper read a path-breaker drug reversing glaucoma. That was all these series of GLP-1 antagonists. So I could correlate. But the interesting part is that when she stopped this, I said 10,000 rupees a therapy for 1 month. When she stopped it, the glaucoma scores start coming back. It means you have to take lifelong to get a reversal of glaucoma. And this was all happening in Hyderabad again in University. And this now, okay, interesting part is another interesting part. Again we went after 6 months and got a checking done. And they said, it is reversing back. I said, should we start with the semaglutide again? We will check with the ophthalmologist. She said, hold on. She Googled in front of me and found that semaglutide and also prostatitis. She said, I am not prescribing it. Now this is a drug where pharmacovigilance is very, very important. They are new class of drugs, very costly drugs for obesity reasons. And recently, exenatide taking nurse has lost life in US because I don't know what the paper is that the same series of drugs when it was taken by her that she lost life. So I will recommend a lot of pharmacovigilance studies over here. And you can see a picture showing profound weight loss after use of GLP-1 agonist, and you can see the pictures that there is a profound weight loss happening. And the landscape, you see over here, nowadays Eli Lilly has come with another brand, Pfizer, Boehringer, Ingelheim, Takeda Pharmaceuticals, and they have revenues now in billions already. And contribution to overall revenue of the company, one single drug, how much revenue it is giving to the company is 20% to 0.2% by Takeda. And global market share is 55-60% by Novo Nordisk. And GLP-1 market is going to go in 2036 is projected to go to 49.3 billion. And see the revenue the companies are going to earn. And now I am learning that they are coming with a 1.5 milligram long term that you can continue 1.5 milligram at a low dose and you can get all the good effects in the body. When diabetes is controlled, you start getting all the organ improvement. Now, another issue which I want to discuss with you here, very important, is types of innovations. Sometimes we have, as a pharma, we are not told of these aspects in our classroom, which one has to understand. There are 4 or 5 types. One is incremental innovation, second is radical innovation, third is disruptive innovation, product innovation, and process innovation. 5 types otherwise. There are more categories also. And out of the major ones are 3 of them. One is incremental, radical, and disruptive innovation. Now, what happens is, over the time period, revenue, efficiency, in radical innovation, sudden change happens, and like I have given you the example of GLP-1 agonist, sudden launch, it jumps the revenue of a company. The incremental innovation is stepwise as you do the innovation, and the disruptive technology beats everything. And this is the chart for Taro. And it's a disrupt the market in totality. Now, there is always a possibility that these type of innovations have overlap. All three types of innovations have an overlap. Now, I give you an example, smartphones, example of incremental innovation. How we started, you can see over here, 1984, this size, slowly, now you have smartphones from 2022 onwards. Now, this is incremental innovation. Every time the company is launching a product which something better, larger memory, larger battery, longer battery, that way changes are happening. Screen is bigger, the screen type is better, you get less strain, all those benefits. Example of radical innovation, same smartphones, they acted as radical innovation at time of COVID. So they were very effectively used in education. You can see over here. And radical innovation is that everyone is having it. Whether that person has no money to eat, but will find a smartphone in the hand or a phone in the hand. That is a very good reality. You can see over here, a rickshaw puller having a phone in hand, and the granddaughter telling grandmother how to use it. The interesting part is it's also disruptive innovation. I know I may not be able to see that day, but maybe in your generation also may not happen, but in next two generations, our necks are going to go like this. Because how much time is spent on smartphones? So this is going to be a reality, and you start getting your neck pains until unless you control your usage of smartphones. How many of you have faced this neck pain? Most of you must have done that. Now, this is disrupting your health. So, typical examples of three main types of innovations. Very quickly I will go. Examples of incremental innovation. First, evolution of different generation smartphones and computers. You have seen, starting from room-sized computers, tape computers, card computers, and now we have the laptops with all the and today, the other day, the CEO of Google was talking about quantum computing. And next is quantum computing. Then added features in subsequent models of vehicles, analog to digital to smartwatches, introduction of automation and robotics, incremental improvements in analytical instrument, mechanization, etc. And you can see over here, the earliest cycle was this one, and this was brought much later, and you can see over here a chart gives how a bicycle, how many iterations it has gone, the simple bicycle. And the same thing has happened in the cars, how it started, and today where we are, very luxury cars with all the comforts. Apple Watch, the incremental innovation that is going in Apple Watch, starting from 2015, not very old. Now we have 2024, and it can detect a lot of your, it can do calls, it can detect your health, everything it is monitoring, and more features being added to every launch. Example from pharma health and technology of incremental innovation, you have new dosage forms, new dosage forms like buccal pills, pulmonary, nasal spray, changing the release sites like mouth dissolving, fixed dose combinations, improved functioning of medical devices, like we have now cardiac implantable electronic devices, drug loading of stents, changing construction material as in case of IV bags from PVC to Viaflex to Viaflow, medication used for diseases different from those which they were originally developed, like GLP-1 agonists, Medications that include devices that facilitate their use, as in the case of injectable like smart computing systems, medication that are integrated with digital applications to improve the management and monitoring, those are dry information. Like Dolo 650 milligram against Chloroxine 500 milligram. So these all are incremental. It was discovered long ago, the mechanism, body mechanism, and the product was brought out, and now the changes are happening in simple ways. So these are all incremental because they have gone to the market and they are invited. That's why I call them innovation. Now, GLP agonist incremental innovation like FDA approval from exenatide, which happened in 2005, and FDA approval was sought in 2012 by AstraZeneca. Then liraglutide then came in 2010, from 2005 to 2010, and all others which are coming out. And tirzepatide is latest one to get FDA approval. So there is incremental innovation. The original was exenatide. Smart infusion pumps, I was surprised I had to go to ICMR, I never seen this, where we had multiple infusion bags to deliver multiple drugs. Here, now we have these devices, they give a very precise dose of drugs which are packed in the infusion bags. Typical examples of radical innovations are LED lighting, electric vehicles, 3D printing or additive manufacturing, blockchain technology was mentioned by our friend Uji, virtual and augmented reality, ChatGPT and AI, ride-hailing service platforms like Uber, Ola, Rapido, online marketing platforms, UPI payments, audio-video and movie streaming services like Netflix, smart cities and all those things. So these are radical innovations. 3D printing, now you can print heart if you have the right bioengineering, and all other, you can see, I went to one of the universities in India in Ahmedabad, Ganpati University. They have created excellent facility for 3D printing. Multiple 3D printing machines brought from all over the world, and they can print anything for you. Blockchain technology is a radical innovation where everybody knows what other person is doing. So it is a totally transparent system. It's not like you are sitting in your cubicle in an industry setup and nobody knows what else is doing. It's a totally transparent system. Virtual reality is a radical innovation, which is gives a stimulated, simulated experience that deploys 3D near-eye displays and post-tracking to give the user an immersive use of a virtual world. We have smart city, which is again a radical innovation, where the transport, people, economy, and whatnot, administration, life, and all those features are coming to the smart city. So it will radically change cities if it is really implemented the way it should be. What are the examples of radical innovation from pharma, health, and technology? Continuous manufacturing, non-destructive analysis imaging, 3D printing of drug production devices, IoT in drug development, supply chain management, modeling and simulation, personalized and precision medicine, cell and gene therapies, CAR-T cell, genome editing, and CRISPR-Cas9, drug discovery driven by artificial intelligence, machine learning, mRNA therapeutics. We have vaccines, COVID vaccine, targeted cancer therapy like Protex, digital health and blockchain in healthcare. Now, already I said guideline has come on continuous manufacturing. And what is continuous manufacturing? Let me explain to you in real sense. This is the future manufacturing of gadgets. In one case, in a room, single room, where you have a big plant, you have a single room in which all in glass casing, the total operations of manufacturing of gadgets is going on. And non-stop, continuous manufacturing. So the benefit is no human intervention over here. No shop floor persons are needed. No BMR. Everything is recorded online. And product, every tablet has the same value. Same assay. Because there is no human errors involved. So another picture is this. So they are actually now available. And these are the excellent innovations. Then non-destructive analysis, radical innovation, testing pharmaceutical directly with without sample preparation they practice. In fact, I have given a few examples only. Now you can see over fully integrated online pharmaceutical tablet titration fat food. Process and technology. So when you are having a continuous manufacturing, you will have to have an online lot of testing being done. There is no finished food testing. So here you can have hardness, assay of blood, everything in a, if you put in the line, manufacturing line. Then handheld Raman spectrophotometer. So you can see over here, there is a plastic bag having a drug pouch, plastic pouch, and you, without opening the pouch, you can test what is inside and what is the assay. Same with the blister. Without opening the blister, you can test it. Even China has launched a spectrophotometer on your mobile, where you can check before eating popping the pill. You can check whether it has a 99% drug or not. China has already launched this mobile with the spectrophotometer. It is known as Chang Wong H2 spectrophotometer smartphone. So here, for what happens for strip? So strips we have the X-ray, which is being used for checking what is the drug inside. So even the headspace, whether it contains the nitrogen or you have created and put an inert gas, we can tell you whether it contains that or not without opening the container. A new chapter in pharmaceutical manufacturing, that is 3D printing drug products. So now we were talking a hospital setting, where ECG machine is there, other monitors are there, there will be a 3D printing machine. When we come to precision medicine, your liver functioning, this liver functioning, this liver functioning are all different. So metabolizing enzyme levels are totally different. So we have to give the precise medicine to him or her. So for this, we need to create individualized medicines. Based on doctors look into the whole data and push to the machine to print tablets, 3D printed tablets. So here you can see the world's first 3D printed pill. And this is a machine which does the job. A small machine and creates a pill. Specific one drug which is very much used and it will be a patient bedside. So modeling and simulation is another radical innovation. There are new terms which are coming into the picture: pharmacometrics, modeling and simulation, model-informed drug development, pharmacokinetic, pharmacodynamic modeling, PKPD, model-informed precision dosing, MIPD, PBPK, physiology-based pharmacokinetic modeling. So their definitions are listed over here, and they are now 95% US FDA drug approvals have a modeling and simulation element. Now my question is, a mother will never allow the clinical trial on a newborn child, or 1 month old child, or 2 months old, whatever. Maybe after some time when he gets big, he or she may allow. But she's never, this is not, the FDA wants, give us a dosing of a newborn. And a lot of newborns are having problems these days. And they're put to the hospital setting and the inpatient and NICUs. So, we have to deliver drugs. How do we calculate the precise dose for that? We have no clinical trials possible there. So in that situation, the only way is modeling. You understand the physiology of a newborn child and also the drug properties, correlate them and find out what is the scalar factor for adult dose to newborn child. So we have done this study for paracetamol and given the scaling factors, which are very similar to US FDA for 3 ages, but we have given for 10 ages in our institution. So successes and opportunity modeling and simulation for FDA. So there are white papers coming out where FDA is talking about this. And this is what we published, PBPK analysis to study the impact of genetic polyformation on NAT2 on drug-drug interaction potential vaccine. These are published in Pharmaceutical Research. Another paper is in molecular pharmaceuticals, physiology based on this another paper by someone else, not our paper. Our paper is this one and another one. exploration of the plausible mechanism capital induced ocular toxicity by using proteomics and quant physiology-based fountain model. Now, ethambutol was eye toxicity. Now, how to determine the mechanism of eye toxicity? I don't have access to eyes to take eye and find out why toxicity is happening and in the living human being. So it is only through the BPK that we prove what is the mechanism of this toxicity. And again, publishing pharmaceutical research. This is someone else, a review article, and there was a two, three workshop summary report. Usually based from biopharmaceutical modeling, best practice for drug product quality regulatory industry perspective. Now this is what we need to teach to the students today. This is very big science. My students, due to this experience and training, they have reached University of Washington, Seattle, and best of the world companies. That is a cell and gene therapist, CAR T-cell, which is again radical innovation. Here we take T-cells from the human whom we want to treat. We make CAR T-cells. This is a T-cell, CAR T-cell in the lab. This is a gene for CAR. CAR is chimeric antigen receptor which we put into the T-cell and we grow them on the Petri plate and then back into it, and they show activity against cancer, specific activity against cancer. So this is very interesting development. India's first CAR T-cell therapy developed by ImmunoAct gets CDSO. So now the CAR T-cell therapy has come to India, but where we are in our academic institutions. So this is the power of startups. When you get it very good qualified, like Mujib has started Insol, and other startups are coming up. A very small startups having best of the 10 scientists working on a specific problem. And this is how they are developing in India. Now research on CAR T-cell therapy rapidly advancing. And FDA approved CAR T-cell therapies, there are now multiple ones available in the market. Now then, there is a genome editing at CRISPR-Cas9 radical innovation, where we cut the gene and insert the bad gene. We want to take a portion of DNA and then insert the good one, and we can heal genetic diseases. So CRISPR-Cas9 technology was a major stir in the field of genome engineering, and it was first described as an editing tool in 2012, and it has made astounding progress in the intervening years. So you can now manipulate genes. And CRISPR, you can see over here that what we do is we cut out the gene part, DNA part, cut out, and delete the gene and add the good one into it, so that all the genetic diseases can be taken care of. Digital health, there are radical innovation, therapeutics, medicine, health. Now we are talking about digital therapeutics, medicine to health. It's becoming integral to prevention, diagnosis, treatment, and management of health and disease. There are a lot of digital approach. Even Chatbot, which we talked in the morning, is all digital device for that same purpose. So, National Digital Health Mission, creating a digital health ecosystem by Government of India, Health Family Registry, Personal Health Record, e-pharmacy, DigiDoctor, telemedicine, Health ID card. The government is trying to even, they have an Aadhaar. You go to the treatment of multiple hospitals, get the testing done in different labs. So Aadhaar project is that all your data will be coming centralized to the government of India. And they will know which diseases you are suffering from. And this data will be accessible to all the health providers. That's how they will see into your screen and then give you prescription based on the nature of diseases you have. And this is Red Health and Vedanta, the UP की पहली 5G ambulance launch की. Now this is a very interesting. This is my son's company, Red Health, which is 1,000 crore company today. And you can see over here, Vedanta wanted them to bring some innovation. And what innovation they did? That doctor in emergency room, to whom the emergency room the patient is taken, He sees the patient in the ambulance on the screen and all his parameters are being transported to and visible to the doctors. Now for hospital, it's very interesting. For a hospital, a patient arriving live is a business. Arriving dead is not a business. So there is so much of the pressure on the company that you really have to bring the patient live. So that's why they have now even bought a company known as a tech company in Chennai, which was giving training to paramedics, nurses and doctors for emergency handling. And they are now going to have, on simulated systems, they are going to train people. Now, the emergency call to ambulance is a person's most vital call of the life. When the person is losing, and the person can die in your front. Your near and dear can die in your front. If ambulance reaches in time, you can save the patient. So what my son has done, he has brought innovation, a lot of, using even AI, that world-class ambulance service is starting. And in Punjab, you'll see very soon these red ambulances. Already in Fortis Mohali, 3 hospitals in Mohali is catering, one in Panchkula is catering to. So the red ambulance is a company owned by my son, and these are promises by the way, and today he could create 1,000 crores. So typical example of disruptive innovation, personal computing revolution, including hardware, software and applications, and digital camera, drones, self-driving vehicles, biofuels, environment-friendly alternate energy like solar wind energy, cryptocurrencies, quantum and cloud computing, commercial space travel, large constellation satellite communication network like Starlink. All these are what we call as disruptive innovations. Now, you have lot of optical fiber put outside your houses for all these 5G networks. When Elon Musk brings Starlink, all this will become redundant. It means it is a disruptive, total disruption. So disruption is groundbreaking, transformative changes that can disrupt existing markets, revolutionary, often creating entirely new markets and challenging established norms. So here you can see these devices no more exist. The other day, these all digital cameras, other things were lying in my home. My daughter came, she's in Goa, she came at home and said, Papa, why you are having all these things and old bad energy? I said, no. I want to show to them, to my grandson, that these were the devices we were using in our lifetime. They will never know until unless they have something digital camera on the front that this also existed. So all these things have gone out and they are no more used. Sony, all other systems. Drones are again a disruptive innovation. So they can supply anything, they can do warfare wherever they are not applicable, and now we are talking of drones delivering the medicines at anybody's home. So when you need, that is what Chatbot was talking about. Quantum and cloud computing is again disruptive. Now we are talking about computing where you have to do calculations which are very large calculations, only can be done through quantum computing and cloud computing. So how easy it is that everything goes to cloud. I prepared slides yesterday and they are automatically they have come to cloud on my mobile. So there's no risk of losing the slide set. Starlink communication, you can see over here, already as of number 2024, there are 6,764 Starlink satellites in orbit, with 6,714 of them working. So SpaceX plans to deploy nearly 12,000 satellites with a population standing that is 34,400. That's why Musk wants to come over to India and sell the Starlink to India, where resistance is there from Ambani. That we also want to set up our own Starlink network. Example of pharma health technology or wearable health monitoring devices. Like inside and out body sensors, genomic data for personalized medicines, medical decisions made with artificial intelligence, AI-assisted medical decisions and drug discovery. Sorry, it's a repetition. brain-computer interface and neurotechnology may enable profound new applications, organ-on-a-chip, micro and nano robots, and gene therapy. Now, very quickly, wearable health monitoring devices, disruptive innovation. Now, this is a very interesting case. The other day, my son came to Chandigarh for 10 hours, and there was a question in CIA. And he was on the panel, and somebody put a question to him, and investors also put a question to him, what is your future next 20 years? Now, very simple, he is in the medical transport. He says one thing is that all the cars are going to be self-driven. There will be no mishaps, no accidents, because all the sensors all over. There will be no road emergencies. Second thing is that you will have all variables on your body. Your mobile, this smartwatch will tell in 3 days in advance that you are going to have a heart attack or you are going to have a stroke. And if there is no stroke will happen 3 days after, but you will reach the hospital in advance. So here is the ambulance need. Then you go by your car and end up in the hospital. So what is the future of ambulance service? So this is a big question investors are putting to him. And it's a very, very going to be a reality. So wearable health monitoring devices, disruptive innovation. You can see how many of your devices you can put on the human body today. And this happened. My son had a patch over here. I said, what is this? It's a continuous sugar monitoring. And coming on the mobile, all the values are coming on the mobile. So very interesting things are going to happen. And when you are in an exercise mode, all these variables are really going to do wonders. Then AI assisted medical research and drug discovery, that is again a disruptive innovation. How artificial intelligence is transforming healthcare. This is a very interesting subject, and this is on the title of the theme of this conference today, AI. And this is now, AI is taught in US from 9th class. And how much we have lagged, that we are not still in higher education, that we are not started to AI. You can see over here benefits of development of artificial intelligence for the healthcare industry, patient data risk analytics, medical imaging and diagnostics, precision medicine, drug discovery, lifestyle management and monitoring, emergency room and surgery, cyber security. So, lot of benefits that are going to come. Now, the magic of AI, which we people have not understood. All of the students who are really on the mobile sets, please look into this slide. Very, very interesting. Here you see artificial intelligence created medicine to be used on humans for first time. So this company from Japan, Sumitomo Pharma Japan recently invented a drug molecule for obsessive compulsive disorder within a year of using Essentia's AI-assisted platform. Yet it takes 10 to 12 years to discover a new molecule. They have well based on Essentia platform, which are the large package software packages which can do modeling and do help you. And all the drugs, all the body receptors which are known, they can interact with that, and they have given a drug within 1 year which are going to clinical trial. Very, very interesting. Now, genomic data for personalized medicines. You have genome data, then we can do gene therapy for genetic diseases again. Now, genetic testing market size, global is at $17 billion, and India it is $60 billion. Who's scope to enter into this field? And it's going to increase. And what we say today, all the girls and boys in the hall, look at the next slide. Janam Patri to Genome Patri. It's a very, very important. My son is a minor thalassemia. And if his spouse would have thalassemia as minor thalassemia, both would have major thalassemia and would have affected the child very badly as major thalassemia patient. And again, blood transfusion for the lifetime. So issue is that you have to evaluate at a right time. From instead of depending on Janam Patri, you have to go for Genome Patri. This is the latest trend. Then brain-computer interfaces, again, this is Elon Musk who is doing these all innovations, which is Neuralink. And your brain-computer will be wired, and your all brain thinking, thought process, activities will be monitored. So brain-computer interfaces and neurotechnology are fascinating fields that are rapidly evolving. And there are a lot of companies working on the Neuralink, not only Elon Musk. Then organ on a chip that you don't do testing in vivo, everything can be tested on a very small chip on which you can test all your drugs. Organ chip recent breakthroughs and future prospects. Let your students read this type of articles. Then micro and nano robots, they can deliver cargo. They can help in cargo transport in cancer treatment, cell therapy, tissue regeneration, thrombolysis, and imaging and diagnosis. Everywhere, and robots inside you, a small robot will be sent into your bloodstream to deliver drugs, to do diagnosis, whatnot. And this is also under development. Enhancing therapeutic delivery using micro and nano robots. Now, gene therapy is again a disruptive innovation. Now, interesting is, there you have therapeutic transgene, which you put into the target cell and which gives you therapeutic proteins, which will be helping in disease amelioration. And genetic diseases are very difficult to treat otherwise. Now, I tell you, on my hand, I have 2-3 boils. Boils you will say or something. Now, this is a disease, I have a genetic disease of some kind, my skin disease. I went to India, I showed to the doctor once, I went for some other reason, I showed the doctor. Doctor said, go to the room number 4, 5, 6, 7, call all the doctors. And he put my hand in front of them, I teach you something in theory. And this is the first patient case that you can see these eruptions on the skin. Now, very interesting. So, had it been harmful of some kind, I ignore it because it's not harmful, but something which is genetic and is harmful and affects your life. So, you have the gene therapy. Now, replacing a disease-causing gene and a healthy copy of the gene replaces a disease-causing gene. Inactivating a disease-causing gene, introducing a new gene, genome editing, that we have already learned about. And what is interesting, world's most expensive drug approved by FDA has multi-million dollar price tag. US $3.5 million per dose. Can India think of coming out with a drug which has a $3.5 an innovation of India, which we can sell to the world? Where we are, where our industry and education system is, where our syllabus is. I was at other meeting the other day, I said you are honoring all your PCI people and you bring them over here in colleges and honor them. Ask them to give a revised syllabus with all these things what I am talking about. What is that? In bold time manner. Don't put them in a high pedestal. Bring them down and tell them that we want a new revised syllabus with all the good elements. High intensity for our students in 3-6 months. That is what our principal should do. And another one, there is a new most expensive drug in the world, price tag $4.25 million. And this is March 2024. Now, somebody has to come up with a scheme, health insurance scheme, which the patient is safe everywhere. He should have a affordability to get this medicine if there is a genetic disease, to save and help in the life. So, friends, a quote by Bernard Dickens in 1979, a medical profession which did not seek to improve means to conquer disease would be condemned for dereliction of its duty. Members of the college will not accept the current state of the medical arts as finite, but feel justified in expecting the development of more effective therapies for illness. and the proportion improved means of preventive care. So, world is watching you, pharmacists. Do develop drugs where there is no cure. So, what we are doing? Are we doing that? That's not dereliction of duty. That if we don't do that. Says that healthcare is better than it has ever been. Cancer survival is at all-time high. Now, this was said in 1979. But today we are much better, of course. Deaths from cardiovascular disease are down by over 40%. Worldwide life expectancy is rising by 5 hours a day. But as the saying goes, that is possible. One recent estimate says that humanity still only has around 500 viable treatments for more than 10,000 known health problems. Think over 10,000 health problems, and yes, solution only for 500. What is the gap? Gap is in the knowledge delivery first what we are giving to the students. It has to be radically changed, disruptively changed. Our classroom syllabus has to be disruptively changed. Don't garland the people at the helm of affairs. Tell them that you have to really come at the table to give us a new space. Which is good class. Thus, an acute need is being felt across the world to speed up access to innovative drugs, devices, diagnostic and digital products to global patients. Innovation trade has always existed in the blood of Indians, which earlier was not emphasized in focus on translation research a few years ago. Dr. Daniel, 10 years ago, was you aware of the word translational? No, I was not aware. And maybe a few years ago that we are thinking of translation research. Now the question is, Indians can do it. Only we have to have a belief in ourselves and prepare students to take the challenges. So it is now resulting in visible societal impact unmet needs. I can give you 3 examples before I stop. I will take the other part sometimes again. The main theme part. India's blockbuster drugs to take on deadly superbugs. It is 5 December 2024. No new antibiotics are coming out from the world labs. But our industry has done it. So they have registered in US FDA new antibiotics which are going to change the whole world scenario and save the patient in the ICU. All ICU patients, I had research that happening in our family. The doctor was saying, please take this person to home because infections will kill him if the disease doesn't kill him. Hospital given infections will kill him. Because we have tested all the antibiotics, now nothing is working. So if you want to save for some long period or long life, only thing is to have new medicines. India is among the countries hardest hit by antimicrobial resistance. In 2009, antibiotic resistant infection caused around 3 lakh deaths. They alone are responsible for the deaths of nearly 60,000 newborns each year. And Orchid Pharma and Moga, they have come out with new, and the other company, Bugworks Research, focused on bugs. They have come out with excellent new molecules, and they are going to be registered in the US. They are done, or they are going to be registered. We Indians can do, yes. Very recently, you might have seen Nanotechnology gift to society for rare disease of what we call as on Mahashivratri. So the gift, they gifted and had an MOU signed with a company. And this drug and this disease is a very rare disease, and it's a skin cancer. And the dose which comes from US and other European countries is 15 lakh rupees. They have produced that molecule, developed a tablet of that molecule, and they have given to company to give free of cost to Indian patients. That company MOU says that we will give free to all Indian patients who have this rare cancer, skin cancer. What more you want to tell? Coming from an academic institution. So my friends, I think I should stop over here, although I have to discuss the steps to promote innovation. That I will come a little later sometime to talk on that. I think you are waiting for the lunch. The initial part on innovation, what the world is doing at this moment in incremental, radical innovations that are happening, and also the disruptive innovations that are happening. I wanted to give you a glimpse of it. And more important, I want to discuss the right definitions of what is idea, what exactly is invention, what exactly is innovation, and what exactly is discovery. Thank you very much.
Thank you, sir. Thank you so much for a wonderful talk. Now I request our respected chairperson, Professor Dr. Anil Kumar and Professor Dr. Vipin Saini, sir, for their concluding remarks. Please, sir.
Thank you, Dr. Siddharth. Sir, your talk is so elaborative and so much informative. You started with your innovation, idea, discovery, which is very, very important to discuss among the young students. And your innovations, discovery, ideas, and examples were very, very informative. You explained also with example regarding penicillin, ether, steroid, morphine, insulin. What is the difference between discovery and innovation? And these days, the government is also promoting a lot of things on innovation, startups. And that's what I think your lecture would be very, very helpful for the youngsters. What is to know what is the difference between discovery and innovation. Your example regarding the incremental innovation, radical innovation, disruptive innovation was so much informative, and it was so much exciting also to know the examples and the development that is going on in the entire globe. I think this information is very, very helpful for youngsters to know different types of innovations, and they are also very, very important for us also to know what are the things that is going on and what is the things left for the young generation to develop further. With these words, thank you once again for your wonderful talk, sir. And I hope the information is very, very informative. Your talk was very, very informative, and you will take a long message in your talk. Thank you.
Thank you, sir. Now I request Director Dr. G.D. Gupta, sir, and Principal Dr. R.K. Naran, sir, to felicitate our chairperson and resource person. Sir, please come on stage. I request Dr. Anil and Dr. Vipin Saini, sir, to please come forward and join. Hello. Please give a huge round of applause for Professor Felicitation. Thank you, sir. Thank you so much for your wonderful talk. Thank you, sir. Now I request Director, sir, and Principal, sir, to please felicitate our respected chairperson, Dr. Anil Kumar, sir. Thank you, sir. Thank you, Dr. Anil. Next, I request Director, sir, and Principal, sir, to felicitate our respected chairperson, Professor Dr. Vipin Saini, sir, from MMU Bologna. Thank you, sir. Thank you so much. Thank you, ma'am. Thank you, Dr. Vinay Chawla, sir. Thank you, Dr. Nitin. Thank you, Dr. Nitin, Dr. Vinay. For (2005), and I am from Jamia Hamza. She has played a role in implementing twice-based credit system and curriculum innovations at Jamia Hamza. She has received several prestigious awards, including ARC Women Scientific of the Year 2023, Women Scientific of the Year 2019, Society for Women.
Hello. Thank you so much for introducing me. This is actually an opportunity for me, and I would like to thank the director and the principal who has given me the opportunity to be here today. And it's actually very difficult. The previous two speakers have made my discussion difficult. I cannot stand before Saranjit sir. I have been a student to NIPER for some time, sir. So it was always I can say good feeling to learn from NIPER, one of the premier institutes. So sir has already both made easy as well as difficult for me to discuss the topic. Though I've been more connected to students, we teach both B.Pharm as well as M.Pharm. Hello. Hello. Hello.
What is it, madam? Kiran madam, jara help करके देना.
Yes, not yet. So I be more connected to students, so I have made my topic or the discussion more student friendly in a way. Sir has given us a very baseline about what is innovation and discovery and the difference between them. I would like to thank Sir for that because it has given clarity to me also on certain things. Since we are all in pharmacy market, I thought why not, because when we say about innovation and all, we don't know about where to get into, because we don't know about the market size. Students in particular don't read much about the pharmacy field itself, though they are in pharmacy, but they don't read anything about when it comes to kind of business. So unless and until you have an idea that what pharmacy market is, you won't go really into it. This is the global market size right now. You can see the Indian market size, right? And the export of Indian market, export of Indian medicines from India to almost half of the globe. We already know, and as we know that India being the pharmacy of the world, and we have proved that in COVID, that we can do wonders. So you need to have the basic, you can say, basic idea that which field you are in. This has already been discussed, right? The various stages of drug development. We start from pre-discovery, going to drug discovery and preclinical studies, then clinical phase, and you get to FDA for approvals, you have labels, and then phase 4 trials. After that, even a lot of things come into picture. Right? So this is the concern where AI is like you use computers always. You talk about computers. We have been listening about these innovations with respect to the computers, because computer is one thing which has changed the world, which has changed the way we work, right? And you have got all kinds of informatics, chemoinformatics, bioinformatics, pharmacoinformatics, proteomics, any informatics you have, and this is the, you can see, huge data that is available, and that has actually led to the separation of various fields based on the information they provide. Right? So when you say computers, it didn't limit to that. So computer was followed by various technology advancements, and AI is one of the technology that has advanced or that has flooded the market with new concepts. In every area of your pharmacy, be it your preclinical studies or your drug development or to market, as has been discussed in previous sessions also, that it is all AI that is taking everything by storm. You get huge amount of data, and that data is being processed by various algorithms and tools, and which give you more accurate results, more accurate details about certain things. So when we say AI, has anyone given a thought what is the market related to the AI? Right? IT market. We don't go into these details as I said. We don't go into the size of the market, because this gives you let's say, a triumph that yes, if the market is there's a lot of scope, and you can see the rate at which they are growing. The IT sector, almost 52%, health IT sector, 52% of the market, fastest growth, and we see India is nowhere. So you have a huge scope over here. AI, we already have this concept of medical writing, right? In India, we have a lot of jobs in medical writing, but where these jobs come from? We don't do it in India only. They all come from abroad, right? From outside India. biomarkers, which we talk about your personalized health and all kind of things, right? The biomarker market is actually booming. Very targeted genetic drug delivery. This has been already talked about, right? Chatbot, stable diffusion, they have been in news for a long now, and actually they are taking a backstage now. So we have realized that it is not only that we require them for writing our Twitter threads or something like that. They have revolutionized, or they have taken over almost every industry in the world. Not only the pharmacy. And pharmacy, we are not, pharmacy has also realized that we cannot work without AI. These are certain examples, right? One was already discussed previously. So there are some examples which were developed, and they have made these drugs with the help of the AI tools. In first case, the 2020, Accenture developed the first AI design drug, right? And you can see in 2021, the DeepMind AlphaFold actually helped us understand the 3D structure of the protein. And it has resolved more than 3 lakh proteins structure. Then in silico medicine started phase 1 trial of AI discovered drug, and there are few more drugs in clinical trials for that. Then few more companies, and FDA is actually taking into FDA has also already accepted these kind of concepts, and there are a number of orphan drugs which have been developed by using AI. Rethinking drug development, the need for change, right? We cannot think developing drug as of today without using AI. And we need to understand why the paradigm shift has happened, right? We already understand the drug development process was a long process, almost 10 to 12 years, but with the help of AI, the time has reduced. What we saw that within a year we could have a drug in clinical trial. Second reason that the cost that involves is huge amount of money goes into the drug development, and most of it unfortunately goes into drain. Because we see that if we are starting with number of drugs, it's only the drug, maybe a drug or two that comes into market. Rest all is waste because they may not cross one or the other barrier that is there for getting approvals for the drug. There was one study that showed that out of 21,000 drugs tested in last few years, only success rate was 6.2%. So it's a huge gap. So that is why to answer these kinds of gaps, the AI has taken over. And it's not me who is telling you or giving you the internet information. It is the companies who are actually the giants in the field. They have realized that the AI is not a kind of option thing. It is now a necessity. You cannot work without AI now anymore, right? So you can see Pfizer has already collaborated with IBM Watson. Sanofi has taken up with startup with Accenture. There are a lot of other companies which are completely doing a project and then selling to the pharma companies. So you can see from this slide, this is again your almost a process of your pharmaceutical business that goes in, that you start from target identification, lead optimization, then go for toxicology studies, imaging and clinicals. So how your AI is actually helping in each and every stage. You can see there is 10% less attrition in identifying target. Because when we say target identification, we have to have a target which is indispensable in that process chain. Right? That's a druggable target. And with the help of AI, we actually screen huge amount of databases and identify a biomolecule which will be druggable. In lead optimization, because once you identify the target, you need small molecules. Small molecules you can have more efficient technology, more efficiently identified by these technologies. We also need less amount of animals to perform clinical studies. Less animals will be sacrificed because these days we have a lot of ethical issues also, so you won't be getting more animals to test your products. So by using these kind of technologies, we actually reduce almost 20% of animal use. And with the invent of technology, you get a lot of images, right? Your MRIs, your CT scans, and huge amount of other scans are there. So you can use those scans and they can be read in a proper manner by these tools and techniques, and you can reduce the identification, or it can identify the patterns that are there and correlate with your process and give you much better information in less time. And then the clinical processes, like it is already explained that with the use of AI, the recruitment of patients, the identification of proper patients, stratification of patients is done much better by using AI technologies, and this leads to result in 50% process automation. That is 50% of the things that are to be done in AI in clinical trials is automated. Automation not only you can say processes and information in smaller time, but also leads to more accuracy. So what do we get? We get revolutionizing data. You have huge amount of data at every stage. The biology, talk of biology, talk of chemistry, or biotechnology, any area you talk, huge amount of data is produced at each and every moment. Here your genetics, genome data, your variables, you get all kind of data with your variables from patients and all. You get your supply chain, blockchain processes, every information is there, and the data is your, every moment actually you are generating data. But what to do with this data and how to use this data is a big question. It's not that easy that the data is there and you'll be able to process it very effectively. It's not a page, it's not a 2-page, it's not an Excel sheet, a 100-cell Excel sheet. It's a huge amount of data, and you need to have some techniques which will actually analyze, identify patterns, and give you some good outputs out of it. Because for computers, it is that's an old thought that garbage in, garbage out. So whatever you give in, you will get out. So the data and the quality of data is very important. And now the data is there, then how can you manage? You need a high-end computing system. You need to have tools like artificial intelligence and machine learning techniques. This slide shows you the artificial intelligence has touched each and every field of your science. When I say science, that means pharmaceutical science. Every field of pharmaceutical science, starting from your drug discovery from identifying your target to actually taking that molecule in the market. And at every step, you are actually using technology so as to have process efficiency. So coming to this term, what we have been talking, artificial intelligence, the machine learning and deep learning. Actually, this Venn diagram tells you about that everything comes under artificial intelligence. Because it's artificial, right? It's not a human-like, but it can do human-like things. So you have a lot of information. It's the art of creating machines that perform functions that require intelligence then performed by people, right? So you are actually training your machines. And machine learning is when you are using a particular data and that program is being trained on certain patterns, right? And deep learning is a subset of this machine learning wherein this behaves as a neural network type, right? It uses GNN as an algorithm which uses, like, it's a kind of brain neurons, how brains connect to each other and process information, it performs it. So when you say machine learning, there are two types of machine learning. Because as I said, I have made it more student friendly. So for students, they need to understand these things, and I have a case study to discuss how you can develop your program or develop your machine learning tool. So machine learning is two types basically, supervised and unsupervised. Supervised is where model is trained on labeled data, and unsupervised is model finds some patterns. You do not actually teach it, and what is to be identified. If there are some informed students, they may have studied or performed QSARs, right? So they have some supervised kind of learning. But QSAR is a very small type. The machine learning uses huge data. Earlier, what we used to do the QSAR, we had a certain limitation that the molecules should be analogous to each other. There should be the biological activity in particular values like ICFCP values or KI values. Then the activity is from the same lab and have better results. So many ifs and buts were there. But when you come to machine learning techniques, you can use the larger the data, the better is the output in that case. So because it identifies the patterns and correlates those patterns to each other. These are some examples of algorithms that are being used in supervised learning and unsupervised learning. You have a lot of algorithms developed. They are further sub-classified based on their different, they can say base concept, like whether they use network or whether they use structure or like. So I have a case study to discuss wherein we have to identify a target. The target should be morbid, druggable, right? Because if it is not a morbid or druggable target, it's of no use. So, but how this machine learning will help us in identifying this data. So you have modern biology, which is increasingly rich in data related to your transcriptomics, that means which gene is transcribing which protein and life. Then proteomics, you have metabolomics, right? And lot of other content clinical images is there. So these data is already there. You need to access this data and develop your model. So what is required is your analytical tool to yield statistically valid, and you can have some predictions for target identification. These are some commonly used. This is for students because where we say normally we see papers and all journals, we don't find much information in a single paper or something like that to actually understand what these concepts are. So these are certain ML tools for identifying gene-disease relationship, right? Because you have to identify the relationship between a gene and disease, then only that gene can be worked on. If the gene is not related to any disease, it is of no use because the gene can be morbid, gene cannot be morbid. So these are certain tools, text mining based tools, network based tools, then ML based features that extraction and prediction. Your number of B-free is there, DigiNet, DeepSeek. B-free is, it uses the drug adverse interaction, what is called drug interaction adverse database of European Union. So it is developed on that. Similarly, your DeepSeek is another tool which uses a further, like a next step. It actually reads papers and underlines the disease that is associated with particular disease. So see, we have huge amount of manuscripts produced every year. Thousands and thousands of manuscripts published every year. But reading all those manuscripts is a big task, it's a challenging task. I cannot read each and every task. Though I remember at my time when I did MPharm, so we used to go to National Library, right, and extract all journals and read one by one, that if any article is of my interest. But this is not anymore. You go to your Google, you give a search and you get the result. You go to your PubMed, you give few words and you get results. All articles of your choice. So similarly, but from those articles even, identifying genes which have particular interaction with particular disease is again a big task. And you may miss out something or miss out one or the other thing. So these actually read each and every article and give you the information about gene and Then when you have identified a gene, the next question comes to any scientist or who works on the area is that whether it is druggable or not. Because if it is not druggable, again, it is of no use. So you can have another a couple of rules, a couple of tools which you can actually help to identify whether these genes identified are druggable or not. Again, those druggable prediction tools are on the basis of your structure and on the basis of your network. Structure is structure of your protein, they express the protein, the small molecule, the information between the interaction of these, and it is on the network that how proteins interact with each other, how protein-gene network is there, how drug-protein network is there. So there are a lot of databases, lot of network databases available which will give you all the information. So these give you information whether the identified gene you have, the genes you have identified are druggable or not. The next important part of the gene comes that whether it is related to the disease or the morbidity, right? Morbidity of the, because not every gene is a morbid gene. So you have to identify genes and set certain criteria before developing your tool or before training your tool. So there are a number of morbidity prediction tools, network-based morbidity prediction tools again, which is on the basis of functional network and machine learning based. There are genomic and mutation-based morbidity prediction tools because genome mutation or gene mutation will lead to the diseases. Then on the basis of these, there are certain others which are computational tools, deep learning-based morbidity tools and meta-classifiers. So when you want to develop a tool or a classifier, you want to train a classifier so that you can work on your area of interest. First thing, you need to define some general rules. You need to compute network topological features, that what are these genes, how they are networked between other genes and other proteins. Then you have to construct it's a database, all the genes which are there present. Then you can have selected features, you can prepare training sets. Because when you have genes, you are preparing any kind of classifier, you have to train it first and then test it. Like I said in QSAR you have. So QSAR first you have to divide your molecules into two sets, training set and test set. On the basis of training set, you used to train your tool, and then test set used to be test that whether the predictions are correct or not. So similarly over here you have to prepare a training set and test set and train your classifier that whether it is predicting right or not. You can have number of features selected, like you can have degree of centrality, that where the exact position of this gene is, like how important this gene is in the particular network, how important it is in producing that particular protein, right? And then how many connections it has, like how many outgoing connections, how many ingoing connections are there for this particular. You can have a number of other related attributes to it, like metabolic interactions, transcriptional regulation, how they are actually behaving in systems, that their position, that their cellular compartments where they are. Then you need experimentally verified databases. Because unless until you have an experimentally verified data, it will be difficult to, you can say, correlate your generated data. Because you have to verify that yes, whatever you are training, it is correctly. For example, I say I am training a dog to pick squares, right? So if I am not training him properly, he may pick only one color of squares. He may not pick all color of squares. So you have to train it properly and then validate it. How will validate? We validate it using some known data which is already proven experimentally. So there are databases which are experimentally verified that these genes cause these kind of diseases. Right? So you experimentally verify their databases and see whether your classifier, what you have developed, is working or not. This will also help you identify the subcellular localization of the human genes. That in which part of the cell this is, the cytoplasm, endoplasmic reticulum, or any other Golgi apparatus. This will also help you that how it is involved in the tissue expression, how it's involved in tissue expression, the tissue expression of that particular protein, which all activities it is involved in there. So once you have identified, you have set your rules, you have identified the genes which you are working on, you need to translate the morbidity database from these genes. So you can have these genes, you can add more genes which are not morbid genes, right? So as to actually confuse your classifier, to give a test to your classifier. So you can add some more genes which are non-morbid genes and can divide it into different data sets. Because the more training it gets, the more accuracy will be there. So you can divide those morbid genes into different data sets. For example, 10 data sets. You can have 10 different training sets and then you can have test sets. So the amount of training will tell you how good your classifier will behave. Similarly, you can use for druggability. You have to have some similar kinds of molecules and then have the druggability data set and divide into small groups of training set and test set to check whether it is predicting properly or not. The classifiers used majorly are machine learning software packages Vega, right? So Vega is mostly like, you can use, there are some more classifiers which you can use. This is one of the classifiers that have been used in this case, and it includes a number of other, you can say, machine learning programs like random tree, random forest, and the JW is one of the basic classifier in the in this Vega, and that is Bypasso environment for knowledge analysis. This machine learning tool. So you can have this tool and develop and train this tool on your database. That means you take your different data sets, train this data set, and then you can say predict for unknowns. So once you develop this, you evaluate this, and for evaluation, these are the parameters. Like in case of your, you see the significance score, R square, is that how this system is performing. So you have we call precision area under curve in this case to actually evaluate how this classifier is performing. You can have also cross validation, right? So you can have 10 normal morbidity data sets and 10 shuffle data sets, and you can mix them and do cross validation for these to check whether classifier is working rightly or not, whether it is providing you the information, because when you are getting the information, you will be doing the next step. So if it is not correctly done over here, you will do the complete experiment wrong. So then the prediction comes, and for prediction you have to set certain rules, and two important rules are there for gene morbidity and for gene druggability. The gene morbidity, that is transcription factor regulation. The more the transcription factor regulations are there, the more important the gene is, the more morbid the gene is. That means it should be transcribed by more than one. Right? Their metabolic and extracellular rule, that means depending upon the centrality of that protein in the metabolic role, how central it is in performing that particular function. If it is central, it is important. If it is peripheral, sort of, it is not that important. But if it is transcribed in more than one gene, it becomes important. Right? So if it is a plasma membrane protein or a protein that works in clustering, so it's an important part. So you have to identify those kind of proteins, genes in your network. Then you need to, if it is your plasma membrane localized, it's a druggable protein, right? Because if you coordinate with your previously identified targets, most of them are membrane-bound targets, right? And then there should be transcriptional regulatory role and enzymatic activity. If there is with this protein related, it's an important in your network. So this experiment was carried out, and it was identified that the regulation in, if it was less than 1 greater than 1, it was true. They were identified 277 genes with this document, and the classifier was done on the data set that was generated on human genes. The value was more than 1, it was true, and it was taken as 277 genes were identified, and the 60 number actually indicates which were falsely identified. So, although 60 genes were identified falsely, but we can see more than 200 genes were identified correctly. Right? Similarly, if it is less than or equal 1, then you have to see the in-betweenness in metabolism. That means if it is important in your metabolism, like the related area, if it is important, then you check for the extracellular. If the value comes less than 1.2, and if it is more than 1.2, it is true. And it is again 269 were identified out of 79 were falsely identified. Similarly, you can see the other parameters which are there in your you have defined your parameters, and you check on each and every parameter whether it is coming true or not, and identify the number of genes. Similarly, for the druggability, you can see if your membrane, if your protein is your plasma membrane bound, yes, it is a true target. It's a targetable gene. It's a druggable gene, and 199 genes were identified out of which 44. Because for every gene, mostly the protein is being expressed. And similarly, you can have other criteria in between. There is regulatory concept is there in between. The metabolism is there. It is involved in metabolism. You can identify whether your genes are morbid or druggable or not. So once you have identified your drugs and identified your genes through this technique, our next job is to identify the small molecule. Right? Again, over the years, a lot of chemistry has been carried out, right? And you have a lot of molecules physically available, and virtually you can design any molecule. So for small molecules, neural network was widely used for QSAR. We know that we had a lot of QSAR studies. The earlier dates back, the 3D QSAR from Grammar, we started. But the question is that, was that enough? No. Right? We have answered already all these questions. So we have now AI, which is actually like breaking all those challenges. You have huge size of data, and you can use lot of network techniques to analyze those data. And neural network is one of the major techniques which is being used to identify small molecules. It has 3 layers actually, 3 layers in a way. That one layer is input layer, the third layer is your output layer, and in between is number of layers. It's not only one layer, but they are set 3-layer tools. So the in between there is a lot of information that is processed, like in our brain, for example. I get a stimuli, and this is an input signal, and I process in my brain. So I don't know how much processing happens in my brain, and then a reply comes, or then a response comes. That is your output tool. So this is an example of 15 data sets, right? If it had been your, like you can say, 15 years back, so this wouldn't have been possible, right? So these are 15 data sets, all done or prepared in different labs, right? So, but you can use a AI model to generate an information for a particular target molecule, whether it will be good or not, or any of the molecules will be good. So you can divide the data sets into 75 and 25 ratio, because you need to first train data, your algorithms, and then you have to test them to actually predict for unknowns. So similarly, the data set was actually sent, and a neural network was used, deep neural network was used. And you can see this from graph that the 15 data sets when used together, they performed much better than the 2 large data sets. Although the 2 large data sets had more number of compounds, but The other 13 data sets performed better than the 2 data sets individually. So this indicates when you have more data, more accuracy is there, because it is able to connect the dots, patterns, and give you better understanding and better results. So this has already been talked about, that it has revolutionized the biomarkers, right? Cancer treatment. You can predict biomolecules from your various samples, because a huge amount of patient data is there available, and using different techniques you can identify biomarkers and have specialized treatments developed patient-wise. These are some examples where you have already drugs identified through AI. As I mentioned, in silico medicine is one company which has already number of molecules in clinical trial, and one molecule for DDRI is in phase 2 clinical trials. So Benevolent AI is already working in atopic dermatitis. Excelsia is working in serotonin 5-HT1 receptor agonist. Recursion Pharmaceuticals again having a molecule in phase 2 clinical trials for cerebral cavernous malformation. Another molecule for neurofibromatosis type 2. It's an orphan drug designated by FDA. Atom has a number of drugs in preclinical stage, so has biotech BTM 162B2. They have number of drugs identified which are in preclinical stage. So this was a brief glimpse about what AI can do, and I hope that with basic understanding of AI, we'll be able to actually do wonders, do better. As Sir has said, that India has potential and we need to revamp our syllabi, and this is one part which must be there, and I'm sure we are working towards like skill-based education. So that will be one thing we actually need, that we need to develop more skills. We are more theoretical today. We need to more skill-based education, and I have come to know that over in here, SFF Pharmacy College, they are doing quite skill-based learning. That's a wonderful thing, and not everybody gets a chance. But I will also suggest, I first thank you all for patient listening. So this was it. I have one request to all the students, which I keep on talking to my students, because as I mentioned, I taught B.Pharm, I teach B.Pharm and Pharm, right? So B.Pharm students are a bit reluctant, like in learning, because the distraction is there in their hands. But you can use this distraction for your, you can say, for your acceleration. Right? This distraction can be your acceleration in case of your whatever you want to do. It's not only the pharmacy, whatever area. I have a simple small suggestion that every 6 months learn some new skill. Every 6 months. छोटा सा एक हफ्ते का कोर्स. There are a lot of courses. I was talking of this company, the first company. I just missed the names. In silico medicine, if you go to the site of this company, it gives you free training, free course for AI with respect to drug discovery and development AI. So you can have any kind of skill you are interested in. Just learn one new skill every 6 months. Thank you so much.
Thank you, ma'am, so much for your wonderful talk. Now I request our respected chairperson, Dr. Vijay Chawla sir, and Dr. Nitish Sharma sir for concluding remarks.
Hello. Am I audible? Hello. Am I audible?
Yes, sir.
Thank you, Professor Akhtar, for your words of wisdom. In fact, it has been an eye-opener for me that artificial intelligence was first coined in 1956. And ma'am has very well explained the various aspects in drug design and development. And she went on to describe the areas which are affected by AI. Not only this, she also mentioned the gaps or the opportunities where we can do wonders using this technique, especially medical writing, mental health apps in market, cosmetics market. So Dr. Akhtar, your lecture was very informative, and we are pleased to learn that there are number of drugs which are coming to market using AI techniques. Not only this, she also emphasized on the need to data integration and evaluation, and overall it has been a very good lecture, and I pity the students who miss this lecture for lunch, because such lectures are hard to come by. She gave us very good examples of the tools that are being used, and she also went on to explain the classifier design, the small molecule design and optimization. Thank you, ma'am, for your wonderful lecture. A big round of applause for ma'am. Thank you, ma'am. Thank you, Dr. Maimuna, for this wonderful lecture on the AI application in the medical field. I can remember a few years back, even the 10 or 15 years, when I was listening to how the AI can be integrated in the healthcare sector. That was the kind of the things which we were thinking it is not possible in the medicine system. But now every day there are a number of products are coming into the market which are developed with the application of this AI and the machine learning tools. And now this is not only the new technology that is become essential for all the healthcare professionals, that without these technologies we cannot think about the product development process. Definitely these kind of the technology are helping in the product development, clinical trials, and the QSAR technology. And your lecture was wonderful, and this was the eye opener for all the students and for all the participants. And I hope your lecture would be the takeaway from this conference from the ISFCON, and definitely the participants will take further information from your resource which you provided in your the slides. Thank you once again, Dr. Maimuna, for this wonderful lecture.
Thank you, Dr. Nitin. We are very thankful to our chairperson also. Now I request HOD, Department of QA and RA, Dr. Balak sir and Dr. G.S. Sharma sir come on stage to felicitate our resource person and chairperson. Please. I also request Dr. Nitin and Dr. Vinay sir to join. Thank you, Dr. Maimuna Akhtar. Thank you so much for coming and give a wonderful talk. Now I request Dr. Balak and Dr. G.S. Sharma sir to felicitate our respected Professor Dr. Vinay Chawla sir from Baba Farid, University of Health Science, Faridkot. Thank you, Dr. Vinay Chawla sir. Please give a round of applause for Dr. Vinay. Now I request Dr. Balak and Dr. G.S. Sharma sir to please felicitate our next chairperson, Professor Dr. Nitin Sharma from MIT University, Noida. Please, sir. Please give him a round of applause for Dr. Nitin. Thank you, Dr. Maimuna, Dr. Nitin, Dr. Vinay Chawla sir. Thank you so much. Now we will meet after lunch. Thank you so much. I request you all